İngilizcesine çok A warm good morning, ladies and gentlemen. I'm extremely honored to welcome you all for this prestigious moment. A pleasant and warm welcome to our honorable dignitaries, respected faculty members, delegates, and of course our dear students to the international conference titled Current Advances in Pharmaceutical Industry and Development, organized by Center for Pharmaceutical Sciences, JNTH, University College of Engineering, Science and Technology, JNTH, and Association of Pharmaceutical Research. Pharmaceuticals is one of the world's most research-intensive industries, generating a continuous stream of new products that save lives and raise the quality of life. The discovery of new drugs has evolved over time from an empirical process to one based to a considerable degree upon a fundamental scientific knowledge. Disruptive technologies and emerging trends such as robotics, artificial intelligence, 3D printing, and precision medicine will impact the manufacturing and distribution of pharmaceuticals. In order to prepare a successful for a better future of healthcare, the pharma industry has to embrace new technologies and put a greater focus on prevention and digital health. We have varied intellects to brief us about this vast subject. Before I introduce them, firstly, a sincere thank you to all our esteemed guests for accepting our invitation and gracing the event. Now, I take pride in inviting the honorable dignitaries onto the dais, volunteers to please assist. Firstly, we have our beloved chairperson of this conference, Dr. A. Jayalakshmi ma'am, Principal JNTH, University College of Engineering, Science and Technology, JNTH, to grace the dais. I request our student to welcome ma'am with a bouquet. Hearty welcome, ma'am. We next have our guest of honor, Mr. Madan Mohan Reddy, sir, Director, Aurobindo Pharma Limited, to honor the dais. Hearty welcome to you, sir. Our next guest of honor, Dr. A. Govardhan, sir, Rector, JNTH, to grace the dais. Hearty welcome to you, sir. We next have guest Mr. Rudra Satpathy, sir, CEO APR, to honor the dais. Hearty welcome to you, sir. Our next guest, Dr. TV Narayana, sir, President IPA, to grace the dais. Hearty welcome to you, sir. We next have our Honorable Chief Guest, Professor Katta Narasimha Reddy Garu, who is currently serving as Vice Chancellor, JNTH, to grace the dais. Hearty welcome to you, sir. Our next guest for the day is a convener of the event, Dr. M. Sunita Reddy, ma'am, HOD, Center for Pharmaceutical Sciences, JNTH, to honor the dais. May I now request the honorable dignitaries on the stage to lamp the light as a tribute to the Mother Saraswati, goddess of knowledge. Ya kundendu tusharahara dhavala, ya shubhravastranvita, ya veena varadanta mandita kara, ya shweta padmasana, ya shankara prakruti devai sada पूजिता सामान पार्तु सरस्वती भगवती निशेष जाड्यापहा. Thank you. I now invite our beloved principal, ma'am, Dr. A. Jayalakshmi Garu, for a welcome address.
Respected dignitaries on the dais, today's chief guest, Professor Katta Narasimha Reddy Garu, Vice Chancellor, JNT University, guest of honor, Govardhan Garu, Rector, JNTH, Rudra B. Satpati Garu, another guest of honor, CEO of APR, TV Narayan Garu, guest of honor from President IPA, Manzoor Hussain Garu, registrar in his absentia, today's conference convener, Sunita Reddy ma'am. Good morning, one and all. At the outset, let me congratulate the convener of the conference, Sunita Reddy ma'am. I am happy and feel honored to welcome you all to the inaugural ceremony of Second International Conference on Current Advances in Pharmaceutical Industry and Development, which is being conducted by Center of Pharmaceutical Sciences in collaboration with Association of Pharmaceutical Research of JNTH University College of Engineering, Science and Technology, Hyderabad. I take the privilege of sharing about the glorious college. JNTH is the first technological university in India, and in the recent past, we celebrated the Golden Jubilee celebrations. on the occasion of the completion of 50 years. This year, we have introduced new courses on computer science engineering and IoT as a minor degree program. We continuously change our curriculum, focusing on the holistic development and in bringing out the hidden talents of the budding engineers by updating the curriculum on par with the industry needs. University College of Engineering Hyderabad has 8 departments: civil engineering, mechanical engineering, electrical and electronics engineering, electronics and communication engineering, computer science engineering, metallurgical engineering, chemical engineering, and information technology. Apart from 8 centers: Center for Environmental Science, Center for Nanotechnology, Center for Biotechnology, Center for Water Resources, Center for Pharmaceutical Sciences, Center for Spatial and Information Technology. All these departments, as well as the centers, offer undergrad, master's program in technology, MCA, MSc, and M-Pharmacy, and apart from that, the doctoral program. And we have a lot of infrastructure with the advanced classrooms as well as the laboratory facilities. Our college is equipped with health center to attend to the needs of the health problems of the students. And we have an indoor stadium with many infrastructural facilities for both for the staff as well as the students. And lot of selections for state as well as national level sports are conducted in JNTH campus. And some of the students could get awards at national as well as international level. Many of our former students have occupied very important positions as successful entrepreneurs, technocrats in the country, and the recent Oscar Award winner, Chandra Bose, who has given his best original song, is an alumni of our college, and proud to say that he is from the department of EEE. I, being a professor of EEE, I proudly announce this. The highest pay package for the last academic year is 46 lakhs, which is offered by the CRED company, which is a startup company. And we have many placements which are going on. Almost 90% of the placements have been completed. We have the students' clubs, which are interdisciplinary and which is numbering unto 13. And there are a lot of programs which are going on with the students' clubs. And proudly, I announce that among the 12 states, we got the Merit program, which is the TechWip 4 funding institute. Technology is a non-linear tool which brings fundamental changes in the society. Today, the technologies are disruptive and bring phenomenal changes like the way we think, act, and perform. It brings changes in the economy in an unprecedented manner than our thinking process and adaptability. The question is how we prioritize our tasks, how we collaborate, how we strategize, and how we innovate and create new collaborative ideas. Hence, creation of IP is one of the important ingredients of the one's own innovation. Even it is changing due to the success of open source movement. Open source movement necessitates the participation of multiple minds, provides a platform for creativity to blossom. But that platform will become success and yield positive result only when we pass through the rigorous test of sustainable factors such as reliability, quality, performance, and sustainability. So many flowers will blossom out of this open movement, but only India needs innovative technological solution in water, energy, healthcare, education, agriculture, aerospace, defense technologies, and other critical technologies. With this, I greet all the delegates of Second International Conference on Current Advances in Pharmaceutical Industry and Develop to chalk out, debate, and finalize the roadmap for their vision. I wish all success in their efforts to create an integrated system or project for finding the solution for the challenges of the world in the pharma industry. Once again, I greet all of you a good morning and thank you for giving me this opportunity.
Thank you for those words, ma'am. Now I invite the convener of this international conference, Dr. M. Sunita Reddy Garu, to brief out about this event.
Good morning. Good morning. Good morning to Honorable Chief Guest, Dr. Narasimha Reddy Garu and Dr. Govardhan sir, Rector of the University, and Dr. Mansoor Hussain sir. Sir is going to join in few minutes. And Mr. Madan Mohan Reddy Garu, Director Aravindo Pharma, and Dr. T.V. Narayana sir, President IPA, who is a big brother to all pharmacy fraternity, and Mr. Rudra Satpathy, he is a CEO, APR, Association of Pharmaceutical Research, and Dr. J. Lakshmi, ma'am, Principal J2H, Engineering, Science and Technology, and my colleagues, Dr. Krishnamon sir, Dr. Ajita, and Dr. Shashikala ma'am, Dr. Uma ma'am, and Dr. Ramesh, Dr. Muralidhar, Dr. Vanita, and other Vanita, and Dr. Padmalata, and Dr. Suresh, and also Mr. Bhaskar and Dr. Srinivas from different affiliated colleges and university. And good morning to all delegates and participants. Warm welcome to Second International Conference on Current Advances in Pharmaceutical Industry and Development. And Dr. Ramkishan has sent me a message yesterday night. He has to go to Delhi on official work, so he is not attending today. And this conference is co-hosted by APR, which has a good reputation and association with medical, dental, and engineering and pharmacy colleges. How I got associated with them in the first, like 2015 or 16, I have attended their conference in Malaysia. After that, I went to Thailand and I went to Dubai as a speaker. Then I was like associated with them from last 5 years. Then we have been conducting the conference last year, March also, during the very severe COVID time. And this is the second time. And thank you, APR, for associating with us. And I feel glad to say that our center has been conducted several seminars, several workshops, several symposiums, refreshers courses, webinars, since the inception from 2007. And we got several fundings like, from higher education, AppShare, Telangana, higher education, AICT, UGC, SERB, and whatnot, Pharma Society, Avnida, and Northwestern universities. Many international and national people has been associated with us. And we have done several things whenever we do the conferences. When we conducted RIPE 2012 and 2015, we had some amount, surplus amount was left over. And that amount we kept as a gold medal, best outgoing student from our department. So far, 7 students has been received. Out of 7 students, 2 of them are doing their Inspire Fellowship on PhD. And we have helped with a surplus amount even to buy some equipment. And also we helped some poor students, like a couple of them like Ramaswamy and Rambabu. Both of them has been settled in their life after we helped them into their career. And for this conference, we invited 5 distinguished speakers, 3 on 16th and 2 on 17th. And the first one is going to be Dr. Rudra. He is going to talk on innovation. And second speaker from Malaysia, she's a Professor Datin, and she is from the head of the department, Faculty of Medicine and Health Sciences, University of Putra, Malaysia. And third speaker is Dr. Sandile Maswaji. She is the Dean and Head, Faculty of Pharmacy, Rhodes University, South Africa. And day 2, we invited Mr. Ishwar Kumar. He is Head and Director, Quality Assurance, Naujan Oncology, Shah Alam, Malaysia. And also fifth speaker, Dr. Virina B. Brand, a consultant, clinical trial logistic biomedicine. She is from the Friedrich Albert Strabe University from the Germany. And I feel very special about this hybrid conference. And the conference received about 458 registrations, of which physical registrations are about 292. Today they are attending. And virtual registrations are 166. And physical oral are 57. Physical poster are 68. Physical listeners are 145. Physical co-authors are 22. And virtual, we have oral 52, virtual e-poster 31, virtual e-listeners are 83. And all of us, all these sessions, we are conducting a parallel sessions, and we are having the, conducting like a 5 parallel sessions in Zoom and 3 physical sessions. One is in this room, 2 in our Department of New IST building. All abstracts will be published in a conference proceedings, and globally various delegates has been attending. One different from India, Israel, one woman, she is already here. She is attending in person, and she is going to present her paper as a first paper. And South Africa, Iraq, Indonesia, Turkey, and Philippines. These are the different participants. They are joining virtually. From Indian delegates, we have various states has been participating. Telangana, Andhra Pradesh, Tamil Nadu, Karnataka, Gujarat, Maharashtra, Uttar Pradesh, and Kerala, and Bihar. And we oral and poster presentations are going to conduct in a 3 physical and 4 virtual in a different Zoom IDs. To evaluate the presentations, we invited eminent personalities for the physical and also for the virtual. You know that, as we all know, that arrangements for the conference is very complex and difficult. I want to thank one and all who helped directly or indirectly for the arrangement of the conference. I thank APR team personally for coming forward, for associating with us. And Hyderabad has a lot of sightseeing places. If any delegate want to visit, we can guide you. And also Telangana tourism is also available to take you to the different trips. We are arranging the lunch in our pharmacy department building, that is called New IST building, for all delegates, which is just 500 meters on your right. And we are also conducting 2 physical presentations in our building. One is in seminar hall and one is in a classroom. And food arranged in the committee hall and 4th floor of the building. You can easily reach there. If you have any difficulty, we have a lot of student volunteers. Can you stand up? All volunteers can help you to take there and enjoy your day, 2 days, and listen to all other participants also. And we are all free to assist you. And we are going to give a special appreciation awards for the best poster, best oral presentations in physical and virtual presentations. And thank you, Dr. T.V. Narayana sir, and thank you, Madan Mohan Reddy sir, and my Vice Chancellor sir, Victor sir, and Jayalakshmi ma'am, and Rudra, and all other principals who came forward and to make this conference successful. Thank you one and all.
Thank you, ma'am. Now I request Mr. Madan Mohan Reddy sir to share his insights.
Respected Chief Guest, Vice Chancellor of JNTU, Sri Professor Narsimha Reddy Garu, respected people on the dais, participants, and students. Good morning to all of you. If you see, for the last 4 to 5 decades, I think we are all witnessed there is a substantial changes in the pharma industry. These are all happened last 4 to 5 decades time. Thanks to Government of India, who brought this IDPL, started in India. IDPL is a government industry. It given a lot of knowledge to the people. Otherwise, we didn't know what is chemistry, what is that really making an API, what is the reactions. Nobody knows. Everyone might be reading or studying in the colleges, but it is fortunate we got the opportunity to work with IDPL. There are so many entrepreneurs came from IDPL, started the industry. Today, if you see what we learned for the last 4 or 5 decades, what we learned is only how to manufacture the product, how to meet the specifications. But other than that, we didn't know in the beginning. Always MNCs used to be there in this business, who used to spend a lot of money for the new molecules, for the new discovery. They used to take a lot of time to develop the products. Their success rate is only 2 to 3%. Once they get the product, they go for the patent and they are there in the market. No one can manufacture in the world other than the MNCs. If you take it 3 to 4 decades back. After IDPL, we got some expertise in this line where we are able to understand what is the requirement, how to go into this business. Finally, we are very successful to go forward, to go into the generic business. The reason is our country or our industry, we can't spend this much of money for the new discovery products. Only we can do is, once patent is expired, we can enter into a generic player. This is what we can do. But always it looks simple. It is not so easy job. Once patent is expired, if you want to go into this generic, we need to show the equivalence to this innovator product. That too, we should not infringe the process. This imprisonment is that is going to be they will suit these companies if you follow their same process. So this is where we got an expertise to do equivalent product in a different process, go into the filing, we got approval, and then we can go into this market. This is what we learned last 4 decades, how best we can manufacture the product equivalent to the innovator product, where we can really be there in the market with the equivalent to that innovators. Today, if you see India, we are the largest player in the world. Anywhere in these regulatory countries like in US or Europe or anywhere, our filings are very high in number. Our manufacturing facilities are too high in number. Our people, whoever is the experienced people, I don't think nowhere we'll get this type of expertise team anywhere in the world. We all got that knowledge. All credit goes to these colleges where, especially in India, there is so many colleges, pharmacy colleges, where students are really focused to learn, then come into that industry where we are able to successfully do all these, whatever that is required for that industry. Today, especially to go forward about that, the regulatory countries, the requirement side, if you see, whatever the expectations are becoming and more stringent today. It is not like that before, where you manufacture the product equivalent and then supply. It's not going to be the end of this process. Today, expectations are too many. Like, give an example of this, I think probably whether you may not be knowing this, even though these products are being manufactured so many years, today the new expectation is some of the impurities like nitrosamine impurities. This is carcinogenic impurities. Nobody knows before. Today, by seeing that lot of whatever developments in especially in the R&D or from the especially the regulatory countries, these expectations, when it comes, we should be able to meet those expectations. That is where our expertise goes into the picture. That is where our industry is capable to see that we comply to that so that we are there in this market, especially for the students. And especially, I think it looks some students are here. There is a huge opportunity for this pharmacy students. The reason is the world market is $1.4 trillion for this, especially for drugs. Our India, we are doing an $50 billion. You know what is the percentage? It is only 3%. There is a huge opportunity for this industry. There is a very good opportunity for the students. Really, there is an opportunity to work. It's not like what I understood is that after completing M.Pharmacy and all, students are selecting some computer, whatever that job and all these things going into this. This is not the correct way of approach. Today, you are in professional. Pharmacy students or pharmacy industry is equivalent to, I think, much more responsible than the doctor. We are all making drugs, quality drugs, we have to make it. So there is an opportunity for you people. Don't look here and there. Then once if you are sincere in your job, there is a huge opportunity for all of you, especially Hyderabad and then especially Telangana and Andhra Pradesh. We have so many industries where there is an opportunity not to go outside or outside country or something like that. There is an opportunity to grow. Especially we have so many R&D centers and then manufacturing facilities, and we are all majority go into these regulatory countries where you will have a lot of knowledge in that. It's if you are manufacturing in domestic, if you are manufacturing in regulatory, I think there is an 500% difference in that the quality of job. It's not just manufacture and then go like this. There is a process where whatever you manufacture today, it is all being recorded. These are all, it goes into the audit trail. Any audit, it can be, you are answerable to this quality of the product. So in this scenario, I think this is the right opportunity for all of you. This type of conferences, you people have to give a lot of importance to this, learn more. Not only that, whatever you have doubts, you have some questions, you must be able to speak to the people who are going to explain you during this course. This way, especially India, what I see, the one concern is you don't speak. This is the one negative point for the teenage students or anybody. Better, I think, speak out. Whatever that you have doubts, please try to take in clarifications. Then make sure that you should be convinced, whatever that been explained by anyone. Make sure that take this opportunity. All the best to all of you. Anytime, actually, especially from Aurobindo, especially I'm telling you, Aurobindo, we started 1986, but we started formulations only 2006 or 7 with $1 business. Today, I think 13 years or 15 years, we reached to $3.5 billion. You see how these are all happened because we are getting in right technical people, pharmacy students, they're all working hard. We are able to achieve this. And also I tell you today, Aurobindo is the second largest prescription in US market. It was there in 21 position, I think some 7-8 years back. Today, we are in the second position. There is no doubt we can come into the front the way we are progressing. We are entering into peptides, oncology, biologics, vaccines. I think whatnot, actually, there is an opportunity for everyone. I think be part of industry. Don't lose your whatever that you are learning, either B Pharmacy or M Pharmacy, and then learning from these conferences. Don't lose and then divert your mind to go into that some different industry and then look for, it looks everything is attractive. Don't look into that. I think be in that and then grow together. Thank you.
Thank you, sir, for all the suggestions and inspiring speech. Now I request Dr. A. Govardhan sir to enlighten us more about this subject.
Thank you very much. I'm not an expert in this domain. All the distinguished dignitaries of the program, chief guest of today's inaugural program of this second international conference on current advances in pharmaceutical industry and development, Professor Katta Narasimha Reddy Garu, the Vice Chancellor of our university, other distinguished guests, Dr. T.V. Narayan Garu, the President IPA, Dr. Madan Mohan Reddy Garu, Director Arabindo Pharma Limited, my colleague Professor Manjur Hussain, Registrar of the university, who could not be here due to some preoccupation, Dr. Ram Kishan, Deputy Drug Controller, Government of India, in absentia, Professor A. Vijay Lakshmi, Principal of UC EST, Mr. Rudra Satpathy, CEO, APR, other distinguished invitees, Dr. Sunita Reddy, convener of the conference and head of the department, directors, principals, Dr. Ramesh, the principal of Viper, all the staff of the former IST, other distinguished invitees, keynote speakers, session chairs, delegates, online and offline, press and media, ladies and gentlemen, a very good morning to you all. At the outset, I congratulate the entire team for organizing this second international conference in the series. I also appreciate the efforts made by the entire team. I also extend a warm welcome to all the delegates on behalf of the university and on my own behalf. As you have listened to the speakers, you have seen the post-pandemic, that is COVID-19 pandemic scenario, the way we lived and the importance that is to be given to the health dimension. So people have realized the importance of health besides wealth. And particularly in the context of this pharmaceutical industry, Hyderabad and state of Telangana is particularly known for the pharma hub. And you see several industries came up and continue to come, several pharma related industries in those lines. And when you look from the research perspective, besides the conventional classical methods and approaches that you do in your R&D activity related to the pharmaceutical domains, there is a need to use the technology to see that your processes get simplified in terms of the time, energy, or in terms of the various resources. So that way you find several recent technologies like artificial intelligence, machine learning, cloud technology, and integration of several other technologies would really help the researchers of the pharmaceutical domain. And if I'm not wrong, the role of the pharmacists those days was like playback singers in a movie, but now you are coming to the front role. You have been, gaining the prominence, though you had the prominence in the global perspective, but not from the Indian and state level perspective. I am sure all of you would definitely gain a lot of importance in terms of the, your role. So that way you have to explore various possibilities to see that how best we can, minimize the, diseases aspects. Particularly you see certain chronic diseases, though, you find the lot of advancement in terms of the technologies, even the lifespan is increased from time to time. And we call 60 plus as young, besides our Honorable Vice Chancellor who is 70 plus, still very young. But at the same time, when it comes to the health point of view, though you have lifespan as the, getting increased, but if you see that your health point of view or healthy index, or if you look at your happiness index, then they are getting degraded or getting reduced from time to time. So this is where we have to pay special attention to see that how best we can address these issues so that our lifespan and our purpose on this planet Earth becomes fruitful. Most of the older generations, if you see, who could live for 80 plus, 90 plus, still could lead healthy lives. But now we are, though we have the span 70 average or 60 plus average, but we struggle a lot in terms of the various chronic diseases and things like that. Particularly how to control these aspects and how to manage them, how to regulate them in the current world of stress. Particularly stress has become a gift for every domain. You work with any organization, you are gifted with the stress as a free offer. So how to control, how to manage besides the usage of the drugs or medicines. So that way, I think this platform would definitely help all the budding researchers and also the experienced people who have come here as the keynote speakers or session chairs. They would definitely share their expertise and also the experience. I'm sure this conference would definitely open the doors of yet unseen world in terms of addressing. Particularly, I always share this on every platform that UN has defined 17 sustainable development goals, SDGs. And if you can really take clues from that, our research becomes fruitful and purposeful. I once again congratulate the entire team, particularly Dr. Sundar Reddy and the other team members for organizing this wonderful conference. I wish that your stay would become a fruitful. Thank you very much for the opportunity.
Thank you so much, sir, for the insightful talk. Now I request all the dignitaries to release the souvenir, the proceedings books of this conference. Same. Thank you, sir. Thank you, ma'am. Now we shall have Mr. Rudra Satpathy, sir, to present a small talk.
Very good morning, everyone. Esteemed dignitaries, revered chief guest, my most respected Vice Chancellor of this esteemed university, honorable speakers, revered guests, honorable and respected professionals from industry and academia, and my students. In this fabulous morning here, we feel glad and privileged to reiterate the announcement of Second International Conference on Current Advances in Pharmaceutical Industries and Development at this brilliant institution that has recently celebrated 50th Golden Jubilee, especially in pharmaceutical capital of India, Hyderabad. Brooded with several geopolitical crises we face today, starting from battle to peace, pandemic to disease, unemployment to economic crisis, we millennia academic entrepreneur believe it's high time to work towards development of science and technology, because basically born in the ages of changing of millennium, we have seen how technology, research and development has impacted our life. But at this forum, I just want to raise the alarm that United States and China alone are contributing towards 50% of global research. But I do regret to say that even of course of development of 75 years, recently we celebrated 75th years of independence, and we have surpassed Great Britain, coming up to us the 5th economy. But the research outcome of our country is only 2.7% across the global research outcome. Niti Aayog recently raised alarm and also expressed his concern, stating that the gross expenditure on research and development of our country is only 0.7%. But if you look at the developed countries of European Union, the GERD, the investment of GDP is 2.8%. And if we keep investing on GERD of our GDP of 2% for continuously for next 3 years, then only India will reach 5 trillion US dollar economy. Well, I had given a very short span of time. Thank you very much. And this is high time to raise up the scenarios, upliftment of colleges. We have more than 2,000 pharmaceutical colleges, huge potency, and successfully our generations have done excellence. Even Vedas and Upanishads have profound evidence of excellencies and outcomes we had. Few lines from Bhagavad Gita is reverberating in my mind. Karmanya bhajikarastu ma phaleshu kadashana. It's the time of Charaka and Susruta, where we excelled in the industry, and the profound universities, the eminent universities of Harappa, Mohenjo-daro, Takshila, and the scripts encrypted in regional dialects like Sanskrit and Pali also do have the evidence of excellence. This is the time to create incubating environment that we are trying to create through this type of conferences activity. Because I strongly believe, even though we are adopting to the change of technology, but we are following the same conventional methodology of research process. Whereas in current generation, we have seen in last decade of doing these all activities and connecting to different professions, there is a metamorphosis, there is a change in psychological, behavioral, and social, cultural, economical aspects. So we have to create multiple circle with research cycle of minimum duration. And I wish everyone to be a part of this conference. I greet everyone. I think this will be a wonderful experience for you. Thank you very much for this privilege.
Thank you for the information, sir. Now I call upon Dr. T.V. Narayana, sir, to address the gathering.
Respected chief guest of today's conference, Professor K. Narasimha Reddy garu, Honorable Vice Chancellor of JNTU University, Hyderabad, and Rector Dr. Govardhan garu, and stalwarts in pharma industry, Madan Mohan Reddy garu, Director Aurobindo Pharma, Dr. Jayalakshmi, Principal, University College of Engineering, Rudra Satpati, CEO, Association of Pharmaceutical Research, and my sister, Sunita Reddy, convener of this conference, and my classmate sitting in the audience, Krishna Mohan, Dr. Ramesh, Srinivas, and other principals, staff, students. Good morning, everybody. Good morning. At the outset, it is my pleasure, it is my honor, it is my privilege to participate in this second international conference on current advances in pharmaceutical industry and development. First of all, my congratulations to Center for Pharmaceutical Sciences, JNTU, Hyderabad, and convener, and supported by Association of Pharmaceutical Research. And it is the need of the day, because everybody is discussing, talking about the research, innovations, everything. But frankly speaking, in India, there is no innovations in particularly pharma industry. So-called 1975, we used to tell 75 years of history. Till today, we have not come out with any NCE, new chemical entity. That is because to come out with one new chemical entity, it takes more than 12 years of research and more than $3 billion of business investment. And we don't know out of the 10,000 molecules screened, whether it may come into market, it may fail at the final stage. So industry is not ready to take up the risk. And even our R&D investment is not even 1%, as rightly told by Rudra. But now after 2004, the scenario has been changed. Every industry is coming with good investment in R&D. More than 15% of their revenue, they are investing on R&D now. We may come with a new chemical entity in future. That's what we are expecting from the industry stalwarts, because Aurobindo is in forefront as per the data, market data. So the companies are striving hard to come out. And one thing I want to remind, the actual research starts at university and institution level only. It is not in the center or R&D center. You take recent developments of Covaxin, Covishield, where it has come. Covishield came from Oxford University, came from university only it has come. Even Covaxin, the virus was developed by our Institute of Virology. So this is the strength of the university. The university is the actual site for the development of research. This was the important thing we have to remain in our mind, and we have to concentrate, particularly the universities. You have to develop, give more concentration on the R&D front, so that the affiliated colleges also will follow. That's what I want to tell on this front. And so many universities came, but I have a special appreciation toward JNTU Hyderabad, because last 1 decade, how the JNTU Hyderabad monitored the institution, how the quality is maintained. I appreciate the university, JNTU Hyderabad particularly, because almost all the Indian university I know, the colleges I know, the problems I know, how the university officials try to maintain the quality education. My appreciation to the JNTU University. And one thing, initially the JNTU Hyderabad started with only PG program. That time also I came one day here and I requested the officials, unless you start with the undergraduate program, you cannot maintain the quality of the education. Then they started. And as per our records of the council, still there is a lacking of the staff. Unless the staff, they feel they are having good support of the university and they are permanent, they cannot deliver the goods. That's what. My request, suggestion, and humble request on behalf of Indian Pharmaceutical Association to JNTU is streamline the staff of JNTU University Hyderabad so that already you excelled in maintaining the quality of the education with the affiliated colleges. And you are coming out with research. Definitely it will be #1 university in India. That's what I expect on behalf of Indian Pharmaceutical Association. Coming to this today's topic, there are 5 essential elements which controls the universe: health, education, technology, poverty, knowledge. But out of the 5 essential elements, health is the first and foremost. That's how the role of the healthcare professionals proved in today's world. That's what I can say. And if you take the technology, pharmacy, it is of the 3 components: science, education, and practice. If you take a triangle, the pharmacy, science, education, and practice, but the center focus is patient. Whether it is technology, whether it is education, whether it's a practice, the end product, beneficial person is going to be the patient. That's what I would like to say. If you take the history of the pharmacy, for thousands of years, we relied upon Ayurveda and Siddha. Thousands of years. And the treatment based on the properties of medicinal plants, animals, minerals. This is how we started our journey as a pharmacist. But 19th century, the drug started produced by chemical synthesis. 20th century, it is the unification of the chemical synthesis and pharmacology. The drug discovery concept has come from 20th century. That is how we developed. That is how the drugs came into the market. Paracetamol in 1956, ibuprofen in 1968, contraceptive pills in 60s, diazepam in 63, insulin in 70, ACE inhibitors in 75. That's how the drugs came into the market. Till 1950, we are import-dependent industry. We don't have the manufacturing facilities. We used to import the drugs. But thanks to our industrialist who has come forward and made us proud that today we are able to produce the drugs #3 in the world. We are able to supply the drugs to more than 175 countries. Thanks to all the pharma industrialists, particularly that too in Hyderabad. It is called as a pharma hub of India. One thing I want to tell all our friends here. Everywhere when we go, sir, we are developing, we are backward in so many aspects. Do you know when the first pharmacy college started in the world? It is in the year 1821, University of Philadelphia started pharmacy. When the pharmacy started in India, it is in 1842 in Goa by Portuguese. So we are not backward. When the first pharma industry started in the world, Beckham, in 1859, Beckham brothers started. And in India, the first pharma industry started in 1892 by Bengal Chemicals and Pharmaceutical Works, Praful Chandra Rai. So we are not backward. Our attitude is backward. That's what I want to tell our friends here. So come out of this. And today, the Indian industry, how it developed from the 1950 import-dependent industry. Now today it has gone to such a stage. More than 23,000 industries are there in India. Out of that, more than 800 are big players. And number 3 in volume and number 14 in value. It doesn't mean that we are manufacturing substandard drugs. We are manufacturing the drugs at affordable cost to the public. That's why the value is less. And at present, we are doing $60 billion business and expected to touch $100 billion by 2025. Exports to more than 175 countries. 50% vaccines in the world are manufactured in India, that too in Hyderabad particularly. And 40% generic supplied to worldwide are manufactured in India. And 80% antiretroviral drugs manufactured in India. And 25% all medicines used in ES are manufactured in India. This is strength of our Indian pharma industry. Thanks to the Indian pharma industry, who are even I am happy, Madan Mohan Reddy sir is here. His contribution is also immense in this growth of our Indian pharma industry. All these things made India as the pharmacy of the world. Whenever we used to attend the FIP conference or WHO conference, when I proudly say I am from India, they used to recognize, yeah, India, pharmacy of the world. That was the credit brought by our Indian pharma industry people. As I said, unfortunately, we are not able to come out with a new chemical entity. In future, we may come out with and fill that lacuna. And coming to the developments, today, current advances in pharmaceutical industry, whatever we achieved today, the position, it is all with our reverse engineering skills, not with our patents or with our intellectual property. But future is going to tell something different, because the technology, today what we learned is obsolete tomorrow. The technology is changing at a tremendous speed. Today what learned is obsolete tomorrow, because the today tablet may not be fit in future. That's what expert says. And recent advances challenged by the current needs. Tomorrow, 3D technology, 3D printing is coming. Artificial intelligence is coming. Cognitive computers are going to take place. IoT is going to rule the future of any sphere, any sphere. So we have to update ourselves, because all these things, latest developments are not covered in our syllabus. And whatever I studied 30 years back, you people are also studying now. Unless you update, attend, you are not going to compete with the future. That's what I used to advise all our pharmacists, because recent development, if you take medical devices, it is our area. We neglected that area. Now IITs are taken over that. The highest patents, every IIT and IIM, they are concentrating on health sector now. But we pharmacists, being a pharmacist, we are neglecting our area and leaving it to somebody else. So my sincere suggestion to all your people is not to just attending and going and forgetting the conference. Take the essence of it, what we can do, how we can involve in research. And even my request to the universities, small country Israel, not even 5 million population. When I sat in the FIP conference, India and Israel, we were together. Every year, they are topped with patents, more than 3,000, continuous 5 years. I asked, what is the success? What is the secret? They told, their strength is university. And the staff were given a task on one particular research. Even the staff leave the university because of superannuation or maybe some other benefit. They are forced to continue the research till the project is completed. Even the PG students, even they complete, when they were assigned the work, even they has to guide and monitor the juniors till the project is completed. That is the success. That's why my request is you follow that model. You be the first, automatically the affiliated colleges will follow. That's what. Why? Even I appreciate Association of Pharmaceutical Research. He has come forward because of the his passion toward the development of the research. Really, my appreciation to you, my congratulations to you for coming forward and encouraging the research, which is lagging now. And drug delivery in future, do you think that the same mechanism is not going to work? It is going to be at gene level. Tomorrow, drugs are coming at personalized medicine. No one drug fits all. That was the slogan of the experts. Because Dolo 650 may not be required. I may require only 500, other fellow may be required 400. It is not. The drug in excess usage is causing all the side effects now. Health related problems. That's why in future innovations, they are going to back to biologicals, back to naturals. Prevention over the treatment. This is the future innovations. And biologics, proteome, biosimilars, biomarkers, bioenhancers. These are all going to change the concept of the drug discovery in future. We have to update, otherwise it will be taken over by somebody. That's what I want to advise our friends. And coming to practice level, telehealth, medication therapy management, public health, and 3D printing, digitization, all these things are going to come. How much we are aware of this? Down the line by decade, these are the changes going to take place in health sector. So concentrate on research in addition to your routine conventional studies. That's what I want to advise our friends here. And top innovations in future is prevention over treatment. Pills for sake of prevention are going to come. And turning back to naturals, that's what we are going to again back to the original traditional practice of medicine. It is going to be authenticated in future. Reverse innovation, which address the huge R&D investment, and nanotechnology. It is the buzzword, everybody knows it. It is penetrating into every field, including diagnosis. Personalized medicine, that's what in future the drug is going to be given as per your gene. That's what personalized medicine. 3D printing, the recommended dose of drug is going to be delivered to you as per your requirement. And artificial intelligence, cognitive computers. These are all the buzzwords going to change the role of a pharmacist in future. Don't think that conventional pharmacist is going to survive in future. Just dispensing and just manufacturing, because your intellectual properties are going to count in future. That's what I want to advise you. I am very happy today. I have seen the speakers, I have seen the topics. It is all going to meet the future requirements, and it will be updated with the latest development taking place in the pharmacy sector. Make use of this seminar, and I congratulate the University of Center for Excellence, JNT University, particularly convener Sunita, and Association of Pharmaceutical Research for coming out with such a wonderful program, which are need of the day. I wish the participants will get benefit out of this deliberations. My best wishes to this international conference. Thank you very much for giving me the opportunity. Thank you.
Thank you so much, sir, for the informational talk. Now I call upon Professor Kadda Narasimha Reddy, sir, to share his insights.
Professor Govardhan, the rector of the university, Sri Madan Mohan Reddy Garu from the Aurobindo Pharma. Principal of the college, Jayalakshmi, Sunita Reddy, Adita, Krishna Mohan, and Rudra Pratap, T.V. Narayan, all participants, etc. Very happy that, this event is being taken place in Jawaharlal Nehru Technological University. I think this is the second this kind of meeting happening in the same hall. Last year we had one and this year we had this. You know that the pharmaceutical industry, as our industry friends and the Association of Pharmaceutical Research Friends and other people who spoke earlier to me enlightened you about the activities of the pharmacist in this country and pharmacy, how pharmacy has been transformed and what kind of research is going on, why it is that important in this country, etc. If you look at the history of the pharmacy in this country, until we had the independence, as one of the speakers just now said, that we used to import the drugs from other countries and we never had a capability of producing the drugs in this country. And healthcare was in a very bad shape. And slowly we have progressed. In the last 75 years, all of you have seen how drastically this particular area has transformed. And this transformation has triggered many things. And this transformation in last 3 years or 4 years after COVID, the transformation is much faster, not only in India, but throughout the world. And there is an urgency and a necessity for discovering new drugs. A lot of research has to go into the drugs. You know that drug manufacturing is a big industry and drug research is another aspect. If you have to find out, if you have to discover one drug, a molecule, you have to work for millions of the molecules, and out of that you will get only one molecule which will be useful for the drug. And this was the scenario earlier. And lot of manpower was needed, lot of money has to go into the research, and that made the, drugs more expensive. But today, because of the fast growth of technology, application of many other areas, now the drug industry has become an interdisciplinary. Pharmacy has become an interdisciplinary. You cannot look at a pharmacy just like a chemistry or any other subject, but it incorporates almost all the subjects. And pharmaceutical engineering is one of the very important area now, because if you have to, once the discovery is made and how it has to be milled, how it has to be crushed, how it has to be, filtered, and so many steps has to be taken place basing on the basic engineering principles. And therefore the pharmaceutical industry, starting from the discovery to getting the end product, the pharmaceutical engineering has become very, very important. Similarly, pharmacology is another area which has become very important, because the pharmacology basically deals with your the drug and what kind of interaction it has on the living organisms. Therefore, every aspect of the pharmacy, you have to go into various aspects. Even pharmaceutical biotechnologists are very important, microbiologists are very important, and biologists are very important, and whatnot, even biophysicists are also very important. So all these things have to be brought into one area where the research ideas can be generated, and where when you take this kind of a help, certainly we will be mastering the area of pharmacy. And it has been a regular and a continuous effort of Jawaharlal Nehru Technological University to bring the technologies into the world And our now paramount interest is to build the pharmaceutical research into a 4. And pharmaceutical research in JNTU will flourish because we are going to have a separate section of interdisciplinary as far as the pharmaceutical studies are concerned. We are trying to bring it in. We are trying to look for the people who can really come and work with Jawaharlal Nehru Technological University in building this kind of a thing. And a new building is also going to come in Jawaharlal Nehru Technological University by December. We are completing that building. That building is exclusively for starting this kind of a programs, interdisciplinary programs, where a lot of research has to go into that. So that with these few initiatives, I think Jawaharlal Nehru Technological University will be in the forefront in research and development as far as the pharmaceutical engineering is concerned. With these few words, I thank all of you for joining here and trying to interact with each one of you and try to gain some more knowledge into research. And your research and your thoughts, future thoughts will certainly pave a way for this country to innovate in new ideas and new products, new drugs in this country. Thank you.
Thank you for sharing the information, sir. Now moving on to the memento presentation. Now I request Dr. A. Jayalakshmi, ma'am.
Sorry, I have forgotten. I want to make one announcement. It is a good news for all our pharmacists. Pharmacist of India is going to be considered as a vaccinator in future. It is a good news. And during the COVID-19, we have approached, Indian Pharmaceutical Association approached the Ministry of Health and Family Welfare. There are 10 lakh pharmacists here. If you want our services, we are ready to work as a vaccinator. But by that time, there was a reply, we got late. Already we have sufficient trainers, and in future, if we want, we will consider as a pharmacist. And I am happy pharmacist is included as a vaccinator in the Government of India, Ministry of Health and Family Welfare. Then we pressurize it through WHO. If government is not in a position to give the training, we Indian Pharmaceutical Association give the training, and we got the approval. From May 15th, we are starting the first batch training in JSS University in Medical College. It is the pilot, as a pilot project. More than 150 students are called for a training. Even we can consider JNTU Hyderabad as one of the nodal center. We can allot this as one of the training center for vaccinator project in future. If you send a letter to IPA, we will sanction, and you be the first in the state of Telangana to give a training of vaccinator. This is the good news for all our pharmacists. Thank you.
Thank you, sir. Moving on to the memento presentation. Now I request Dr. A. Jayalakshmi, ma'am, Rudra Satpathy, sir, and Sundar Reddy, ma'am, to present a memento as a token of gratitude to our chief guest, Professor Katta Narasimha Reddy Garu. It's a request to our honorable chief guest, Professor Katta Narasimha Reddy Garu, to present a token of appreciation to all the dignitaries. Starting with Dr. T.V. Narayana Garu. followed by Mr. Madhumohan Reddy Garu. And to Dr. A. Govardhan Garu. And to Dr. A. Jayalakshmi ma'am. And next, Mr. Rudra Satpati Garu. Continuing, Dr. M. Sunita Reddy Garu. I also request Dr. G. Krishnamohan sir and Dr. Ajita ma'am to accept our token of gratitude. I also request Dr. K. Suresh Kumar sir, Associate President HR, to accept our token. Thank you, sir. Thank you all. Now I request Dr. A. Ramesh sir, Principal Viper, to deliver out of thanks.
Good morning, all of you. At this outset, I would like to profusely thank all the people here who have made this a Golden Jubilee function. Basically, I would like to thank the Bhishma Pitamaha of the JNTU, Professor Kattana Narasimha Reddy Garu, for being here despite his busy schedules. Secondly, I would like to thank the smart and ever active dynamic rector, Professor Govardhan Garu. Above all, words won't be sufficient to express our thanks despite a family health issue. We have Dr. T.V. Narayana, the big brother of the pharmacy colleges across India. Thank you so much, sir. And our heartfelt thanks to the guest of honors, Dr. Madan Mohan Reddy Garu from Aurobindo Pharma, and the principal JNTU H. Vijayalakshmi Garu, and the CEO Raghu from APR. And I would like to share the special mention of all the persons who have been involved in this function, majorly from CPS, Dr. Krishna Mohan, Shobha Rani ma'am, Ajita ma'am, and all their team of volunteers, the staff of UGC, and a team of persons who were involved in making this a grand success of this magnitude within a short period of time. And lastly, but not the least, the woman entrepreneur, the woman dynamite of our pharmacy profession, right? I would like to thank the ever dynamic active lady of JNTU, Sunita Reddy Garu. Give her a big round of applause, right? Who has been very active and making us taking forward. So thank you one and all.
All the delegates, we shall have a small tea break and be back in 10 minutes, please. for an online session. After the session, we'll have a dance by our CPS students. Please stay back.
Okay. Hello, everyone. Can you hear me?
Yes, ma'am, you are audible.
Okay, right. Wait, I couldn't see my thing. Right. Okay, right. So, hello everybody. It's afternoon in Kuala Lumpur in Malaysia. Before I begin, I am Professor Sharida from University Putra Malaysia. And wait a minute. And I would like to thank the organizing committee, in particular, Mr. Kartik Raja, for inviting me to deliver my talk for this conference, Second International Conference on Current Advances in Pharmaceutical Industry and Development. The Association of Pharmaceutical Research and the Center for Pharmaceutical Sciences, Jawaharlal Nehru Technological University of Hyderabad. So hello everyone. Before I begin my talk, I would like to introduce my university and our location in the map. Some of you might be familiar with my country. Some of you might not be so familiar. So I would like to share with you. Is the slides changing or is it still on the first slide? I have changed into the second slide. Is it moving or not?
We can see only the first slide, ma'am.
I'm not sure why. I have already changed the slides.
The second slide is visible in a smaller screen.
Yeah, I'm not sure why that is. It looks like the slides is not moving.
Yes, ma'am. It looks like the slides are not moving.
Yeah, so what should I do?
You can find the slideshow option at the bottom. So you can put it in slideshow actually.
Yeah, I have already put on the slideshow. My slides is moving, but I don't think your screen is moving.
Ma'am, like you can see the second slide. We can see the second slide, but in a smaller screen. Can you please click on that second slide? Yeah, now it is moving, ma'am. Now it is moving.
Now it is moving.
Yes.
Okay, good.
Yeah, now it is fine, ma'am.
Okay. So, okay, good. Right. So, okay, but I cannot go to slideshow. Okay, probably not a slideshow. Okay, I'll go to the, okay, you can see my second slide. So this is where we are located, okay? And this is Kuala Lumpur. So my university is known as University Putra Malaysia. Yeah, but I cannot have the animation if I cannot put the slides. Why can't I go to slideshow?
Ma'am, at the bottom right corner, you can find a small option called slideshow. When you click on that, it will be visible in the full screen mode, ma'am. Yes, that one, ma'am.
I have done that for my screen. Press F5. F5. F5. No, it's not doing that. Because it looks like there's no animation in here. Okay, anyway, I'll just make do with whatever that I have. Okay, so this is part of the picture on how our university look like. It is of a green campus. Yeah, so, and this is some quick facts about our university. It is one of the premier public university in with an array of disciplines, and our ranking is moving up the ladder. And in 2023, we are 123 in the world according to the QS ranking. And our niche area is in agriculture, forestry, animal science, pure science, and engineering. And having said that, we have various other area that we embark on. Okay, so we have about 16 faculties, 2 schools and 2 centers that offers about 141 different programs. Teaching courses to undergraduate and postgraduate studies. And it involves research and services. And this is where I am located, which is the Faculty of Medicine and Health Sciences. Okay, so that is all about my university. And for the title of my talk, it's about our group that is doing research in drug delivery system in precluding the progression of tumor. Okay, and our group consists of chemists, that look into the synthesis of the material and develop and also doing some molecular screening and also scanning. And I am in the biomedical sciences, which actually took the synthesized material and tested in vitro as well as in vivo. Okay, and before that, the material will be characterized and we will actually test for the drug uptake, localization of the drug, and we test on mechanism of anti-cancer and look at the molecular target as well as gene targeting. Okay, and then at the same time, we do some toxicity screening for acute as well as subacute toxicity. So for this study, we actually divided our work into purpose of application and then development of materials and look at the molecular target pathway. Okay, and then the purpose of the application of our synthesized material is for therapy, for diagnosis, for imaging, and then we also have development of vaccine as well as treatment for prophylactic treatment. Okay, and our material, the system of the delivery for the drugs is lipid systems. We also have polymer system and we have inorganic hybrid system. And then after the synthesis of the material has been done, then we look at the uptake of our system, whether it is actually targeting the cancer tissues or whether it will have some effects on the healthy tissues. So the modulation of the pathways will be investigated, and then we actually test our drugs and our synthesized system into the in vivo animal model. Okay, so we have various faculties involved with our work and various institute within the large institution. So these are where the location of the research being conducted. So basically during the tenure of my work, tenure of my service in this institution is actually looking at drug discovery and development. So as you all know, the drug discovery and development is actually a tedious work and it's a tedious process which are bringing the drugs to market to be used by the society, by the patients as well as the populations. So I'm sure everyone is aware that the drug discovery would involve the drug design up to the testing of the drugs as well as natural products or anything that you synthesize upon in vitro and in vivo testing assays. And then when the mass screening and the potential compounds has actually successful in the preclinical testing, then it is approved to be tested in human, in which the clinical trials will take place for another at least 5 years. And then after that, then if it is approved and if it is confirmed of the safety aspect of the compounds, then the drugs can be registered to be approved and to be used by human. Okay, but all this process is actually demanding vast testing procedures and a lot of costs for the pharmaceutical industries. So within the duration of the drug development, it may reach of a total of more than 10 years, which could be between 12 to 15 years. So despite this vigorous testing and despite a huge amount of money being invested in the drug development, and sometimes it could reach up to $1 billion, at the end of the day, it could be a successful drug and it could not be a successful drug. So it can cause some failures in the development, and it can cause a lot of money to the pharmaceutical industries. So nowadays, what pharmacologists and scientists has done, the drawback of testing material actually generated a large number of compounds in the libraries, which allow the purpose of repurposing the compounds as well as the drugs. Because some of the materials could actually be able to answer in vitro as well as in vivo testing, and to be used for some activities or some pharmaceutical claim, and yet it may not be suitable activities in human. So this drug is actually being able to validate for the activity of drug repurposing. So the tested compounds can actually be used to actually cut the cost as well as cut the duration of the stages of drug discovery and the development. Okay, so as you can see from the slide, the number of compounds tested in the first stage, it could be up to thousands of compounds, and after all the stages which I have mentioned earlier, it could only left with only one compound. Okay, so in order to get this compound, billions of dollars has been invested, and you are left with a few thousands of compounds which cannot be marketed or cannot be used. So strategies to adopt to identify a new drug candidate nowadays, you can actually cut costs by doing the drug repurposing methodology. So after having done all the stages, the screening as well as the preclinical testing, so the libraries of drugs has increased, so there is a speedy way to find new drugs. So what we did is actually to go back and screen for the drug libraries and then access on previous literatures, so that we can actually repurpose the old drug for new treatment for a common disease, new treatment for a rare disease, or new treatment for a neglected disease. So these strategies has been seen while in the past 2 years, it has been confirmed, it has been shown working, especially during the pandemic, when we need a shorter time for drug development, and we need a costing of the financial purpose to actually bring the drugs quicker into market. Okay, so in slide 16 that you can see, this is another example of the strategies of using the drugs, tuberculosis drugs, okay, isoniazid, rifampicin, ethambutol, pyrazinamide, to reduce the cholesterol level in patients. Okay, and this has been researched worldwide to see whether treating tuberculosis with lipid-lowering drugs, statin, will actually reduce the action of will actually reduce the cholesterol. Okay, and then this is another example of drug repurposing in which metformin, which is an anti-diabetic drug, has been shown to significantly reduce the risk of tuberculosis in patients with a hyperglycemic condition. Okay, and then metformin has also been shown to improve tuberculosis treatment outcomes among the diabetics. So even during the pandemic, various drugs that has not been brought up into market, or the drugs that has been used in market, but that drug has been repurposed for other purpose as well. For example, you got an antibiotic that has been used to actually tackle the side effect of the novel coronavirus COVID-19. So drug repurposing is actually something that has been practiced nowadays by the pharmaceutical industries in order to recycle and also to reuse the drugs that has been approved earlier, and yet it is not being used for the purpose that we want to use today. Okay, so what we have got in our laboratory is to actually test on the drugs for delivery system that is to treat cancer. And we believe that by having the nano delivery systems, the targeting for cancer, it's much more potent and it is much more specific. So as everyone knows, the cancer treatment is actually having some drawback in which the chemotherapeutic agent is actually reducing the efficacy or reducing the drugs treatment for cancer patients. So 10 most common cancers in Malaysia is the breast cancer, colorectal, lung cancer, as well as the head and neck cancer. Okay, and according to the Malaysian National Cancer Registry, the breast and cervical cancer has been the leading cause of death for Malaysian women who are contracting with this disease. Okay, so what we have found that the percentage of cancer is actually doubled and is expected to rise by 2040. So what we found that the rise is actually more than 10% rise. And from the study that has been reported, the rise is actually due to the changes in the lifestyle factors that affect the weight, in which that the diet is not of healthy diet, increase in alcohol intake, reduction in physical activity, as well as the smoking behavior in our population. So even though our country is actually having a good healthcare system and a good socio-economic program, but the screening, the response to the screening of cancer is still poor. So the total of the cancer cases in 2018 is actually rising, and it is actually up to 44,000 cases back in 2018. And the total number of cancer deaths is actually plunging to 27,000 cases 2 years ago. And due to the fact that the screening and also the diagnosis of cancer is at a later stage, then the trends of premature death in cancer is actually in the situation that worries the country. Okay, so the increase in risk and the increase in the rate of death is actually worrying. So what we feel that it is necessary to do is actually to modify the chemotherapeutic agent. Because sometimes when patients undergoing chemotherapy, there is so many systemic side effects in which it is due to ineffective targeting. And when the chemotherapeutic agent is actually targeting the healthy cells, then patients having experienced all the side effects. So what our group has done is actually to develop nano therapeutic system to actually target some cancer cells, and we have passive system as well as active delivery systems. So the active system is the passive system is to the passive system. What is important is that the passive system is able to target the specific cancer cells through the utilization of the EPR effect, in which is known as it is due to the leaky vasculature of the cancer tissues or the tumor, and it is known as enhanced permeability retention effect. And then that actually makes sure that the delivery system is actually concentrating on the tumor. And the active targeting, we do have targeting of the specific receptors that is highly expressed on the membrane of the cancers, and ensuring that the drugs is actually specifically on the disease tissues. Okay, so our research is actually taking the chemotherapeutic drugs that has been approved by the FDA, and at the same time, we are also looking on the natural products material that is exerting on that is exerting cytotoxic effects on the cancer cells, and we repurpose the drugs by targeting on the disease tissues. And our essays is actually following all the guidelines, the International Conference on Harmonization guideline. We also follow the OECD guidelines, and we also follow the Good Laboratory Practice guidelines. Okay, so the drugs or the delivery systems that we have is liposomes, inorganic layered double hydroxide, the zinc layered hydroxide, the zinc aluminum layered double hydroxide, zinc magnesium layered double hydroxide, graphene oxide, iron oxide, metal organic framework, and zeolitic imidazolate framework. So after synthesizing the materials, we would actually characterize the nanoparticles so that our nanoparticles is having the favored physico-chemical characters to ensure the quality of the material. So various parameters was conducted to investigate our nanomaterials in order to ensure the loading efficiency is good, and then there will no impurities associated with it, and physical state of the material is associated with high efficacy of our drugs. Okay, so we have various services as well as we have various facilities within the campus to actually conducted the characterization process of nano delivery materials that we have conducted. Okay, and after synthesizing, after characterizing the physico-chemical properties, we would actually test our materials in the cells to ensure that the level is the activity is actually better compared to the one without the delivery system. And after we are happy with the screening process in vitro, then we will conduct it on our animal model. So the animal model that we have developed so far is the hepatocellular carcinoma model in mice, and we have published the model in +1 paper back in 2018. So when developing animal model, it has to be precise, because this material is actually to treat the organ which is inside the body. And at that time, when we conduct the animal testing, we are only allowed to do chemical induced cancer treatment. And we didn't do xenograph induced liver cancer. So it has been a good study, and it has been a good observation that we made through the induction of hepatocellular carcinoma in the mice, and that study was conducted for about 12 weeks. For about 12 weeks, 3 months to 4 months. And our treatment is actually, our induction is actually giving all the symptoms that is required to confirm that the animals actually develop liver cancer, and we ensure that the cancer is actually exists before the treatment can be done. And the existence is being conducted before treatment is done, compared to the one that is not being induced. Okay, so marker of the liver cancer is to be investigated before treatment is done, so that you are able to claim that the treatment is actually reducing the liver cancer in the animals. Okay, so analysis can be done through biochemical markers as well as physical observation on the animals. And at the moment, we are in the process of developing in vivo deposition of our metal organic framework, and our work is actually stuck at the moment because we are not able to secure a grant for now. But we are hoping that if there is any chance that we can form collaboration with anybody, that we can actually go for grant application, because in vitro testing has shown that the compound that we developed to treat lung cancer is actually deposited in the lungs. So we hope to be able to test our material in the animal model to see if the application of chemotherapeutic drugs through inhalation is giving a better option compared to the conventional chemotherapeutic agent that will give various other side effects to the animals or to humans. Okay, so I'm sure that all of us are aware that at the moment, the purpose of having a drug delivery system is being accepted by the drug company, that this system is actually good to target the cancer cells much better and much more specific compared to the one without the systems. So FDA has actually approved various drug delivery systems back in the early of the year, end of the year 1995, and various other drugs is actually coming up that is approved to be used as drug delivery system. Okay, so with that, I would like to say thank you for giving me the chance to deliver the talk, to actually introduce our group, and also to introduce the project that we are doing in University Putra, Malaysia. So thank you very much.
Thank you, ma'am.
You're most welcome.
The session is open for queries. Please, participants, if you have any questions regarding the session, you are free.
Hi.
Rabbit ka?
office.
Hello, ma'am. Hello. Hello. What are the challenges in computer-aided drug design?
Hello. Yeah. Okay, that is a very good question. Challenges in computer-aided drug design. Okay, this is involving technology and it is involving in the course. Whether the place that you work on whether it's a university, whether it is a research institution, whether it is pharmaceutical industry, big or small, whether they would want to actually look into the computer-aided drug design. I would say that the advantage would be huge, but the challenges is whether the technology is, we can actually pick up the technology. So like somebody like myself, probably I'm not an IT savvy, to be honest with you. Okay, so I need to actually learn about bioinformatics, and I have to learn. But to me, computer-aided drug design, it's a much more quicker if you are able to actually get hold of the technology, because you would actually just sit in front of the computer and then you should be able to actually look at different orientation of drugs to the receptors, whether it actually match, then you go and do the wet lab. Because computer-aided drug design will actually cut on the things on the screening process. Yeah. So am I answering to your question?
Yes, ma'am. Thank you. I have few more doubts. Yes, ma'am. Okay. Thank you, ma'am, for your answer. I have few more doubts. Nanoparticle formulations are free. The nanoparticle formations easily get exposed to free radicals. That means the API get contact with free radicals and it leads to toxicity. How to minimize the toxicity of the nanoparticle formations?
Okay, that is another good question. Okay, nowadays, well, even oxidative stress, even free radicals, without the nanoparticle, your body is actually producing constant free radicals. So how much that it can, the body can tolerate? That's number one. Number two, when you give a material that is able to increase in the oxidative stress, but you are giving it to cancer cells. So in a way, if you look at a different angle, it is actually killing the cancer itself. because the stress is actually in the cancer and you are actually boosting the host immune system to actually kill the cells. So you can actually look at into different angle. So it could be good, it could be no good. But then nowadays, scientists is actually looking at increasing the oxygen radicals in the cancer to boost the immune system of the host.
Okay, thank you, ma'am. Thank you. Your talk was very informative and inspiring. Thank you once again for that. I'm passing mic to my friend. He have some doubts.
Hello, ma'am.
Hello.
Nice to hear your talk, ma'am. Ma'am, I have one doubt. How to optimize non-degradable target to degradable target?
How to optimize non-degradable target
To degradable target.
So what is the degradable and non-degradable target that you are looking at?
Sorry, ma'am. That
What
is
do you mean? What do you mean?
Drug-able to non-drug-able target.
Sorry, say that again.
I will repeat my question, ma'am. How to optimize non-drug-able target to drug-able target?
How to optimize drug delivery target?
Drug-able target, ma'am.
I'm repeating the question again. How to optimize non-druggable targets to druggable targets?
Non-druggable target. Non-druggable target to druggable target. Okay. I'm not sure, but I can try and answer. Okay. So how to optimize the non-druggable target to druggable target, meaning how to optimize the molecular target to make sure that it is targeted by the drug. Is that what you meant?
Yes, ma'am.
Yes. Okay. Now, the one that we do is drug repurposing. Yeah. So drug repurposing, in a way, we would know the mechanism of action of the drug. So in a way, we would know which molecules that is targeted by the drugs. And at the same time, we would know the receptors on the, for example, cancer tissues. What are the receptors that is there? So we would actually attach to the delivery system in order for the transportation is actually specific to that particular receptors, and at the same time it will target to the disease tissues.
Yeah, thank you, ma'am. What I understand from these students, the drug is not able to target, means it is not able to reach that site. So they want to modify the drug to the target.
Okay, yeah.
So that can be done by different methods like liposomal nanoparticles.
Yes.
Or neosomes or ribosomes, any kind of material or any kind of novel drug delivery system, they can do it and target to that tissue. Okay?
Yes.
Okay.
Yes.
That you answered your question? Okay. What is your question? No, that won't work. Tell me. Shappu.
Madam, he was to ask, his question is that, for example, let's say if you take Alzheimer's, you will see the Alzheimer's disease, for example, if you consider amyloid protein, right? So it is misfolded form, right? So that leads to Alzheimer's formation, right? So you can consider that targets of this Alzheimer's disease, mostly if you see, they are non-druggable, right? You cannot make a drug molecule using that particular protein or a macromolecule, any macromolecule. So how do you convert? How do you optimize that macromolecule into non-druggable target into druggable target?
Because at the moment, we only work on specific delivery system to actually target the receptors on the membrane.
Okay.
Okay, because they have to make a more lipophilic.
Yeah. Okay.
Okay.
Yes.
Yeah.
And then we make it, we can make it more lipophilic. We can actually have a ligand on the receptors. Yeah.
That is the answer.
Next, one more, madam. You mentioned about cancer, right? Cancer. You can deliver a drug molecule to the cancer cell lines. But in our body, if you see cancer patients, they are having highly acidic cells, right? Acidic. So how do you overcome that? They will degrade the drug delivery system, right? If you take any formulation, they will degrade the particular formulation. How to overcome that?
How to overcome that? Okay, that is a good question. Another good question. So usually what we have, we ensure that the charge of our molecule is also like having be able to actually target the acidic condition. So usually how we develop is that so that the charge is actually positive, so that it is able to be attracted to the negative and acidic condition and to be able to be in the cancer microenvironment.
Okay. Thank you, ma'am. Thank you. Thank you for joining us from Malaysia. Thank you. A big applause to you.
Thank you. Thank you.
Thank you. Okay. Yeah. Can we start the next one or shall we? It's 1 o'clock. So dance. Okay. CPS students come forward. Have a dance so we can have one more speaker for half an hour and we'll break at 1:30. Quick. Come on, quick. Girls, where are you? Come on, come on, come on. Oh, you are there. Want to have connection? Put it, come on. Thank you, ma'am. Thank you. We'll see you again. She's still there. Jahnavi. Jahnavi. Jahnavi. She's okay. She can tell her. Okay. Yeah. Can you call the next person, next speaker? Dance ready to join out the one. Join out.
वो बटन निकाल के उसको फोन करना तो हाँ पहले चेक किए तो आ रहे ना हाँ कुछ सेटिंग नहीं किया हाँ चार कुछ सेटिंग मत किया स्पीकर में नहीं आ रहे नहीं तो उधर नहीं आ रहे हाँ हाँ इसको वो जूम वाला
We'll proceed on to the next session. There is a small like network issue with the dance.
अब एक बार चेक करेंगे అప్పుడు వాళ్ళకి చూస్తున్నారే అది ఫస్ట్ చెక్ చేసి ఫస్ట్ చెక్ చేసింది మళ్ళీ సెట్టింగ్
ఇక్కడ మాట్లాడుతున్నా వినిపిస్తుంది చెప్పండి ఇంకా సెషన్ మరి
So moving on to the next session, we have Professor Dr. Sandile Maswazi Kamanga, Dean and Head, Faculty of Pharmacy, Rhodes University, Makanda, South Africa. It's about just 10 minutes.
సరే hello
Good morning,
Can you
Professor.
please
How
speak?
are you?
Dr. Sandile, we can't
Good
hear
morning.
you. Please unmute your mic. Dr. Sandile, can you hear us?
I can hear you.
Jahnavi. Dr. Sandile, can you listen to us? Are you able to hear
Yes.
us?
I can hear you.
Wow. Yeah, I was. Yes, sir. We can see the messages, but we are unable to hear you. Can you unmute your mic?
Am I audible?
ఇందాక మొబైల్ కనెక్ట్ చేసి పెట్టారు भाई एक बार ऑडियो जैक निकाल के रखो
You're getting the code? Miss, did you get it?
That guy, no.
Huh?
He's graduated.
Hello, check. Check, check.
Good afternoon, everyone.
Good afternoon. Dr. Sanile, can you hear us?
Hello, hello.
Dr. Sanile, can you hear us?
I can hear you.
Yeah, thank you. Thank you. You can go ahead and give the presentation, Dr. Sanile.
Thank you so much. But I can't share my video. It says you have to give me access.
Yeah.
It says the host has stopped it, but I'm able to start. Thank you.
Thank you. Yeah, it's all set. You can go ahead, Dr. Sanile. No problem. We are fine.
Okay, thank you so much. I am Sandile Kamanga from Rhodes University in South Africa. We are one of the 9 universities that offer the degree in pharmacy, master's in pharmacy, PhD in pharmacy. I am also the dean and also the head of the faculty. I'm not sure if you can still see my slides.
We are unable to see your seat. Yeah, now it is visible.
Thank you. And I also want to send my special thanks to the organizers of the conference I am very excited to be part of the conference. I am going to briefly talk to you about taste masking of a drug by an ion exchange resin using an electronic system, an electronic tongue system. The outline of the presentation is briefly presented here, and I'm also aware of the time that you have allocated me. I will spend some time on introducing the topic and also looking at the anatomy of the tongue and the mechanisms that are related to that. And I will also share with you one or two methods that we have used in analyzing one of the products that we have prepared using an electronic tongue. That it's going to be in a form of results, and I hope I'll be able to end there. When it comes to masking the taste of bitter tasting medicine, there are a number of techniques that one looks at. You can add sweeteners, you can look at microencapsulation, you can think of using inclusion complexes, and you can also use ion exchange resins. pH modification is also one of those. Or you can use nanocarrier systems, specifically the liposomal approach. And in our studies, and the presentation that I have today is going to be mainly on ion exchange resins. And we have also done studies on microencapsulation. And it's also important to understand the evaluation of the masking technique. What are the processes that are out there? One may think of panel testing, and obviously that's got issues. You can therefore go the instrument route, thinking of spectrophotometric methods, and also using an electronic tongue. Looking at the sensation of taste, it is pretty obvious that one, if you want to receive medicine from a pediatric formulations and also geriatric formulations and also other adults as well, active pharmaceutical ingredients, they come in different organoleptic properties. Some are salty, some are bitter, and the tongue in its nature is able therefore to pick up some of these. And there is also an ongoing research that looks at other sensation of taste, looking at the pungent and also the coolness of a product. But the first 4, 5 are the ones that are mainly we encounter in most of our products. And just going straight to what the tongue looks like, in the tongue, how taste organization comes about. There are taste buds which are found in the tongue. And to a lesser extent, you find them in other regions of the oral cavity. And these taste buds, they've got a lifespan, which is about a quarter of a month, or also a third of a month, 8 to 12 days, just depending. And these, they differ from each and everyone's individual. And it's also important, therefore, that when it comes to oral tasting, we appreciate as well that there is going to be personal subjectivity. And it's also when it comes to doing studies, therefore using human panels, you are going to get subjective results instead of objective results. And it's also using animals for these studies. There is no similarity in flavor perception between humans and animals. One also needs to go further to think about the ethical consideration when you are going to subject a panel of individuals tasting medicine. And these may also be time consuming And it's a costly exercise because it needs to be a proper conducted studies that would elicit or give out a reliable and also predictive results. Hence, in studies, therefore, it's important to start thinking about using an electronic palm. I've already spoken to the type of techniques that we have used in our laboratory. We have used microencapsulation. We have also used ion exchange resins. And this is what I am going to be looking at. And the advantages of the ion exchange resins, I've indicated them on the slide. I'm not going to go through them because of time issues, but it also improves adherence in terms of taste masking. Once you have taste mask, the bitter tasting medicine, and the patients, therefore, who are taking the medicine will adhere to their regimen that they are given. So therapy, therefore, it's going to be improved in terms of efficacy and also just by improving the organoleptic properties of the API, one will be able to improve therapy in this way. So the anatomy, therefore, when it gets to the tongue, there are three types of taste buds that one look at. You've got the fungiform taste buds, the circumvallate taste buds, and the foliate taste buds. And these are located in the taste buds. These are the different taste buds located in the tongue. What they do, they transduce taste stimuli, which means that they change the taste stimuli into electrical signals, which are then transferred into signals for the sensory nerves. And there are three important nerves that are found in the tongue. And the cranial nerve 7, the cranial nerve 9, and the cranial nerve 10. These three nerves that are associated with the taste, they all play an important different roles for the transduction or the electrical signals to be transferred. With the three that I've presented on, they are also associated with them 5 different cell types. Type 1, type 2, and type 3. Many articles, books, they actually talk about 3 distinct cell types. The 4th and the 5th one, which is the basal cells and the neuronal cells, there is not much that have been really looked at on them. These cells, they transmit or they transduce taste into different forms that one can actually understand. And we are going to be spending more time, therefore, in looking at the 3 distinct cell types, type 1, type 2, and type 3, as I've indicated, even though there are 5 different or distinct cell types. Where do you find them? I've already spoken to that. And I am going to jump this slide and go to the next one. In here, I've just shown a slide. This has been taken from an article written by Stephen Roper and Nurapi Chandari, where they've actually shown that the taste buds are organized into 3 main types according to their functions, which I've already indicated in here, the different types and how they actually work. You can see the type 1, the type 2, and the type 3 cells that are found in the tongue. Going forward as well, associated with these cell types, there are neurotransmitters. We spoke about the types buds, we spoke about the cell types. Now we are looking at the neurotransmitters. 5 important neurotransmitters that have been put up that are involved, therefore, in the taste stimuli part. And early suggestions they show that ATP is a transmitter for taste buds, where you see immunostaining for P2X, purinoceptors, nerves associated with taste buds. And these are very important receptors that are found in the taste buds. And you are also going to see that the 5-hydroxytryptamine 5-HT receptors are also involved in here. So there are studies that have been done as well that show that using cellular biosensors identified TAP3 cells as the source of stimuli secreted by the 5-hydroxytryptamine. And there are also, we start looking at the GABA. In this case, is the GABA aminobutyric acids. They are also involved and in the synthesis and also the stored in and the release of the type 3 cells, the type 1, type 2, and type 3 cells. And also one looks at how, in addition to secreting ATP, the type 2 cells also secrete acetylcholine, which other studies have suggested that early findings, these taste buds are also rich in acetylcholine. Colleagues, once we have the anatomy of how transduction of taste signals work, I'm just going to jump this slide so that I'll be able to complete in here. The Neuroscience second edition book, it actually gives us a very, very good part of how the whole mechanism of taste happen. It involves calcium-gated channel, voltage channels in terms of how the transmission is happening. It's quite a complex space where there are second messengers, and there's also sodium channels, polarization, depolarization, G proteins that are also involved in the whole mechanism of taste transduction. In here, we are looking specifically in the interaction between a drug molecule or an API that is beta in taste, and all that is going to be managed with the mechanism. So any issues that are related to taste buds, that are related to the type of cells, that are related to the neurotransmitters, it may, to a great extent, therefore compromise the quality of life of a patient when taste is really a big issue when it comes to a formulated preparation that is out there. So we then start thinking about it in terms of improving non-adherence, and masking of a beta molecule therefore becomes very important. And I've already spoken to these slides in here. Our group at Rhodes University, we have looked at microencapsulation, where we use captopril as an API of choice. Our work starting from 2010, 2011, 2012, we have published in the Journal of Microencapsulation, where we have done that in just looking at microencapsulation. We've got current studies where we now look at the effect of the microencapsulation in terms of the efficacy or the effectiveness of the taste masking. So that's one of the studies that we have looked at, and we've done evaluation of the dosage forms as well. And we've gone a step further now, looking at captopril in combination with hydrochlorothiazide, and also in taste masking. The work as well has been published. And now we go straight to an electronic tongue. So this is just the steps that we have used in the microencapsulation. Our current work, therefore, it looks at using ion exchange resin, using captopril and DOEX 66 as a basic for the ion exchange resin to produce resinates. that we have then used to prepare a dosage form that we have tested to see if taste of the captopril has been changed. The choice of an ion exchange resin depends ultimately to the API of choice. Captopril is got an acidic thiol, it causes a bitter taste, therefore a basic and ionic resin was chosen or selected for these studies. We first had to wash and purify the resin that we had before we started preparing our dosage form. So this is the processes. We have shown that in articles that we have, and I'm going to show you the article that we have actually published on that with a group from Poland. The electronic tongue that we used for this work was in Poland. We have ours now currently, an electronic tongue. So the one that we use, it was a sensor array which had 16 classic ion selective electrodes that were specific for the drug that we have chosen. And in this particular case, you have to have the reference electrode, which was the silver chloride one electrode was used as a reference. And we have the tester electrode and also the reference electrode that we have in the electronic tongue. So the differences, therefore, in the signals over time will be able, therefore, to give us information about the effectiveness of the taste masking that would have been done in that. Okay. So the potentiometric sensors that we used in the electronic tongue, whilst in the first instance, you see that it is, when you buy them, they're quite expensive. But once you have a system in place, they become simple to run. So the ion selective electrodes in this particular case, they represent the largest group among the potentiometric sensors. There are also voltammetric sensors, but the ones that give good results for our studies that we have looked at are the potentiometric sensors, and they gave satisfactory results. We're able, therefore, to evaluate these results and process them, these results using principal component analysis, which is the PCA in here, and I'm going to show you the kind of results. In here at Rhodes University, we've recently purchased our own electronic tongue from a company in France, Alphamos. It's an S3 electronic tongue that we use for many dosage forms, starting from not only pharmaceutical products. We are going now to the wine industry. We'll be going to the dairy industries and also the confectionery industry, and also the quality of water. We can use it for that. So I'm just showing you the one that we use here. The one that I have used in Poland, the picture was not clear, so I didn't show that one, but we do have it. So how it actually comes about, you get chemical images of all samples at different time intervals. And using the PCA, the principal component analysis, it will give us coordinates at where the pure API is situated in the coordinates. And the pure API without the microencapsulation or without the resonates, you actually are able to measure the distance between the API and the new formulation. And that gives us, therefore, the ideas as to how, in the terms of the two-dimensional plot, for all the formulations that we have tested at different time intervals. So the ratios that you see in the colleagues, it just speaks to the ratio of the API to the resonate or polymers that have been used. So the potential taste masking properties of the complexes were found, therefore, to be dependent on the ion exchange ratio of the captopril to the ion exchange ratios. So the differences then that you have, if I had an hour, I was going to be able to explain more in terms of the trend that was actually we saw with the work that we have done. On analysis, there was no correlation that we actually saw in terms of the dependence of the concentration of the resonates to the captopril. The correlation was complicated and a nonlinear dependent and it was also nonlinear dependent. And this was confirmed using the electronic tongue and dissolution testing. I didn't show the dissolution testing results in here, but I just wanted to show you how from the PCA and the software that we use, we are able therefore to see, to locate in this particular case, look at the captopril, the red dot where it is, and then we start looking at the different concentrations of the resonates, where the distance from the plots, you'll be able to see that. This work we published in 2020 in pharmaceutical development and technology. I did this work with my colleagues from Poland, Prof. Wesley, Prof. Kozanowska, and Prof. Siosek. Those are the ones who are the other group that I work with for an electronic tongue when it comes to taste masking. The clear separation that we have seen from those plots and also the Euclidean distance, it just shows, therefore, the kind of effects that the ion exchange resins have. So these chemical images alone exhibited the highest value for the PC2, which is this one here, are the different electrodes that we use. We are able as well to come up with the specific electrodes that will be specific for the API, and also that will give the highest sensitivity for the work that we do. And when you start looking at it in terms of the images as well, you need to be able to see, comparing this in terms of the 5 minutes that we have here, 10, 20, 30, up to 60 minutes, it relates to the dissolution studies that we have for the product. So after 5 minutes, we can actually be able to track based on the PCA where we have located each and every drug resonate combination is. So what you can actually get from this part, therefore, is that there was an effect in this case of masking of the bitter taste of the API. So the Euclidean distance calculation for pure captopril using the PC1 and PC2, those are tables that we have shown in the article. If you just go to the article, you are going to actually have most of the work that we actually show in that part. So we take into consideration these chemical images after 5 minutes for each concentration, after 10 minutes for each and every concentration. These exhibited, if the one that you see exhibited the largest distances to the chemical image of pure captopril of 7.6 and 7.7 using arbitrary units for the PC, suggesting that effective taste masking may be possible for this test formulation. The drug resin complexes, after tasting them, they show that the bitterness of the API was not there. It was essential for us to purify and activate the resins when we started to remove any impurities and residuals that one will start with when it comes to the resins. Sorry. In other studies that we have done, on top of the electronic tongue, we've used spectrophotometric methods where we take the drug resin complex, we take it together with the buffer at 6.8, we mix the two, and then we take the concentration. So these are other studies where we have used clarithromycin in the same way. What we get in there, we get a threshold concentration. We determine spectrophotometrically, or whether it's HPLC, the concentration of the drug that will be released when you do dissolution studies. If the concentration, or when you look at dissolution, the concentration of the drug, it's lower at pH 6.8 or 6.7, 6.5, that it tells us that in the mouth, the pH saliva, when you are unable, or when there is a reduced concentration in dissolution, it tells us, therefore, at that particular concentration or at that particular pH, the drug release is going to be minimal. So From the tongue perspective, therefore, the taste of the bitterness of the drug is not going to be realized or felt at that point. So in closing, I just want to bring into perspective that the human panelist, you get good results, but the results are subjective. To get objective results that one may be able to use, instrumentation play an important role where you can start using an electronic tongue, and it's going to give you nice chemical images that one will be able to effectively interpret. You can also use spectrophotometric results that are collected at different time points for specific pHs, and there you will be able to see a reduction in the concentration of the drug that will be released at different time points. I want to acknowledge my colleagues, Dr. Ntemi. I supervised him for his PhD. He worked on clarithromycin, a bitter drug. I want to acknowledge my colleague, Dr. Chikukwa, who did a lot of work on captopril, and we have published these studies on the articles that I've just indicated using electronic tongue. A co-supervisor to this project, Professor R.B. Walker, and I also want to take this moment to thank Rhodes University, and also to take this moment as well to thank you for inviting me to the conference, and also to be part of the panelists. And also, which is lastly, I want to thank the organizers, APR and the Center for Pharmaceutical Sciences, UPG CST, Jawaharlal, and the University of Hyderabad for the opportunity to speak, and also to all the other guests and everyone who's has joined this meeting this morning. I'm happy to take questions starting from the first slide, and also to share some of the articles that we have presented, articles that we have out there. The limit for me was the time. I wasn't able to really project everything that we do in 10-15 minutes. Thank you so much.
Test marking of drugs by ion exchange resins using electronic study system. And thank you, Dr. Stanley. I know it is already midnight for you. And thank you for joining us and enlightening us. Thank you very much. Any questions from the audience? Okay. Thank you, Dr. Stanley.
I greatly appreciate. I'll stop sharing.
I appreciate you. Thank you. Okay. Yeah, girls, come on to the stage to have a 5 minutes dance, and then we can proceed to lunch. Just go out of this auditorium, turn on your right. Just 500 meters you walk, we'll get a Center for Pharmaceutical Sciences, and you have a coupons. Please give the coupon and take your plate, okay? And enjoy your lunch and disperse to the rooms. Whoever presenting here, come back. Rest of them, you stay back. Their seminar hall and also classroom in the same building. You can have your presentations. Virtual already started. They're going parallel, okay? Okay, girls, come on to the stage. And these are all my students from the Department of Center for Pharmaceutical Sciences, and they're going to rock. Okay, thank you.
देववरीतं वराहरूपं देववरीतं परस्मितवदनम् वज्रदन्तधर रक्षाकवचम् प्रणवालय परिपालय परमेशी कमलालय श्रीदेवी कुरिपिंसवे करुणाम्बुरासी धींतारा धींतारा जगुलतो प्राणमे नाट्यं చేసే గతులతో నామశతం మూల నతులతో నాపైన నీ చూపేలా శశివదనే శశివదనే స్వరనీలాంబరి నీవ అందెల వన్నెల వైఖరితో నీ మది తెలుపగరావా అచ్చొచ్చేటి వెన్నెలలో విత్సంగాలు నవ్వగనే గుచ్చెత్తేటి కులుకు సిరి నీద అచ్చొచ్చేటి వెన్నెలలో విత్సంగాలు నవ్వగనే గుచ్చెత్తేటి కులుకు సిరి నీద
యమునాలో నల్లనయ్యకై ఎదురు చూసెనే రాధా ప్రేమ పొంగుల పసిడి వన్నెలే వాడిపోయెను కాదా వేయి గడిచెను పగలు గడిచెను మాధవుండు రాలేదే రాసలీలల రాసురానిదే రాగబంధమే లేదే బృందావనం నుంచి కృష్ణుడు వచ్చాడే యమునా తీరాన ఉన్న రాధను చూసాడే దాని కుడి భుజం మీద కడవా దాని పుట్టపురై కలమరియా అది రమ్మంటగా నరసనియా దాని పేరే సరంగ దరియా దాని ఎడం భుజం మీద కడవా దాని చెంటువై కలమరియా అది రమ్మంటగా నరసనియా దాని పేరే సరంగ దరియా
ఎగ్జిబిషన్ చేస్తాడు అయిందా ఫుల్లో ఉంది మన ప్రాబ్లం ఏం లేదు మనకే నెట్ మన ఉందా మా నెట్ ఇంకా అదే చేయాలని ఉంది నాకు ప్రాక్టీస్.
అది Peru's door. Mimosa. Mimosa, mimosa.
by defining what is medical instruments.
Comfortable Hindi or English. I told Kannada. Tata Play. They are our. We are customers. We have to give service based on our language. He's calling and telling the language you want. You are told Kannada. I am
ఐదు మినిట్స్ 10 minutes అది కూడా చెప్పాలి నువ్వు మధ్యలో వచ్చినాక టైం 1 minute 5 minutes ఉంది సార్ అది కూడా చెప్పలేదు ఫస్ట్ ఏం చెప్పలేదు ఫస్ట్ చెప్పలేదు you have the 10 minutes you have to complete it slide పెట్టాలి అది కూడా చెప్పలేదు నేను స్కిప్
Yes.
అలానే ఉందా మోటెన్ చెక్ అలానే
చాట్లో ఏదైనా ఏదైనా
Good afternoon, all. The virtual presenters in the main hall are requested to move to your corresponding presentation halls, corresponding breakout rooms. I repeat again, the virtual presenters who are in the main hall are requested to move to the corresponding breakout rooms. We are having 4 halls, Hall A, Hall B, Hall C, and Hall D, where the parallel virtual presentations are going on. So if you are a virtual presenter who is there in main hall, please move to your corresponding breakout room. Thank you.
is eligible to national health insurance. The government is taking care that every citizen will be able to get medical care, but the medical care is given to him by 4 sick funds. There are 4, everyone can choose a sick fund or a health services. They changed their names. Once they were named sick fund, but now they are named health service, like in the United States, you know, health maintenance organization. So in Israel also health service, not sick, but so why we have 4? Because of medical history. The state of Israel was established 1988. At the same time, the British left India and Israel. At the same time, they went back to Britain and left us to have our own country. But the medical services started 120 years ago before the state. Before the British came, there was the Ottoman Empire. But because Israel is the Holy Land and pilgrims arrived to Jerusalem all over generations, even the Middle Ages, you know, the Crusaders, everyone came. And many people came because they wanted to see God. They believe that God is staying there. So they came and they saw this poor and dirty city. So they already came with mental problems, and then they became more sick. And the church wanted to take care and established hospital. 120 years ago, there was a Christian hospital in Jerusalem, and this hospital still exists. So the Jewish communities in Europe say, no, we can't send our sick people to the church. So they also established a hospital. So we have very old and very big hospitals more than 100 years ago in Jerusalem. And in fact, they supplied services to all over the country. It was mainly charities, it was free. At the same time, young people who lived in Europe who suffered from anti-Semitism, you know, in Russia, in Germany, they discriminated the Jewish people. So young people say, enough is enough. We are going back to Zion. It's written in the Bible that God promised the land to the Jewish people. We are leaving Europe. We are coming to Israel to establish our own country. So they started 100 years ago. My grandparents arrived 150 years ago, also because of their religion. They say, we want to live in the Holy Land. Why to suffer in Russia or in Poland? We want, you know, there are always massacres of Jewish synagogues. So they wanted to move, and they came to Israel and established farms, agriculture, and they also needed medical care. So what they did, they make a fund. That's why it's called sick fund. Everyone has to put money. They hire doctors, nurses, everything. And they say, we have a health fund that if someone is sick, they will take care of him. So this is the reason that nowadays we have all these sick funds that are stronger than the governments are. You know, they have more seniority. They have the doctors, they have the clinics, they have hospitals, they have everything. So when the government was established in 1984, they said, okay, we have someone who supplies the services, let them go on. So they went on for 50 years. They didn't stop them at the moment because of political reasons. Why? They belonged to the trade union of the workers, and it was a party. And they, in fact, collected tax. They have also their own funds. They collected money. I was a student. If I need medical care, I had to be a member of the trade union. If you are not a member of the trade union, we are not eligible. So only 30 years ago, we have the national health insurance. And what happens that all these charities and organizations were out of money. They were the manager, but the government had to keep the money. They didn't have enough money, nor the charities, nor the sick funds. So 30 years ago, the government said enough is enough. We are now controlling the money. We will give the money. They will give the services. So, in fact, And let's go now to pharmaceuticals. Now there are maybe 20 institutions that buy pharmaceuticals. First of all, we have the 4 CFAs. Now we have 5 hospitals of the charities, of the churches, and of the Jewish communities. As I told you, the Jewish community established synagogues, established hospitals. More than 100 years ago, they still hold them. They don't have money, they pay nothing, but they hold. They are the managers. I mean, they are as if independent. So they also buy medicine. Now, as for hospitals, we have about 12 hospitals that belong to the government, which they inherited from the British. The British also funded. And there are about 6 hospitals that belong to the sick fund. Now also the army needs medicines. So altogether there are at least 20 institutions that buy medicines. And then the government said, oh, there is one producer. As for pharmaceuticals, there is only one producer and 20 different institutions that buy medicines, so it's a monopoly. If it's a monopoly, we have to control the price. So the government is controlling the prices of medicines. Time? Oh, numbers. Sorry. But just to put the story short, the government is controlling the prices and the sick funds, they have to manage about the substitute, what to buy, how much to buy. This is our number to show how they are sensitive to the prices. Okay, thank you very much.
Yes, thank you.
So I just wanted to show you the quality. But if my friend has to carry a knife to open it, and if you put drops to the baby, and then all the bottle comes out, maybe it's cheap, the price is good, but the quality is bad. And maybe the medicine is correct, but the envelope maybe is not done well. And the sick fund is also taking care of it.
Please do send me here. Here, hospitals.
Thank you so much.
Yeah, to take this subject up.
Oh, thank you.
Right here because
Oh, thank you.
negative. And then PPT ఉంటే లోడ్ చేసుకుంటాం.
Hello. Yes, sir. I can start.
5 minutes.
They have to change the slide. Good afternoon, everyone. This is Pranita from Gokaraju Rangaraju College of Pharmacy. Today I'm here to discuss about the transcriptomics in medical research. Epitranscriptomics is the field of molecular biology that includes all the biomolecular modifications of the RNA within a cell. For decades, scientists have knew that RNA was decorated with chemical modifications, but forgot about them. The creation of new fields, new tools about the modified RNA now had emerged into the transcriptomics. There are different types of mRNA modifications. Those includes ribosomal RNA, messenger RNA, and there is a huge diversity in messenger RNA. We can find that there is 3 to 7% of total RNA by mass in mammalian cells. And there are different types of mRNA modifications, and more are yet to be discovered. mRNA base modifications involves attachment or removal of methyl group at a particular position on the base. One such modification is M6A modifications that includes methylation of N6 position, resulting in the form of M6A. The methyl transferase 14, that is METTL14 and METTL3 are involved in this process. M6A RNA modification regulates the gene expression in a biological process like RNA. RNA splicing, nuclear exposure, stability, and degradation. Enzymes control the fate of RNA and thus the protein production. This is very interesting to know that the enzymes are controlling the fate of the mRNA. Those are the proteins that are produced, and so some of the enzymes are writers, erasers, and readers. Writers are METTL3 and METTL14. Erasers are FTO and readers are YTHDF. Role of mRNA modifications in human diseases. A to I RNA modification in treatment of cognitive impairment. This is a neuro disorder where generally the unmodified glutamate receptor allows the calcium influx to that is leading to the cognitive impairment. Here the individual adenosine base mRNA are modified into inosine with the help of ADAR, that is adenosine deaminase acting on RNA. Thus, the CA plus influx is restricted and results in healthy neuron. The role of M6A methylation is regulated in the pathogenesis of brain injuries. Thus, it can be treated during the brain diseases. And FTA plays an oncogenic role in acute myeloid leukemia. Acute myeloid leukemia is essentially a disease of dysfunctional differentiation. Here, generally, the healthy people bones are filled with hematopoietic stem cells, but
people suffering with leukemia, the stem cells are haywired, where the proliferation, they are proliferated and displace the other blood cells. And they can't be differentiated into mature and into normal WBC. This causes leukemia. In 2016, there was a study shown that tissue samples taken from people suffering from myeloid leukemia displaced high levels of enzyme FTO that is discovered as M6A eraser. The importance of FTO in leukemia targets FTO signaling by selective inhibitors may represent a promising therapeutic strategies to treat leukemia. M6A in cardiomyocyte proliferation. In M6A RNA modification, YTHDF is reduced from binding to the protein YAP, and the protein YAP mRNA translation is destabilized. ALKH-B5 eraser demethylates M6A YTDHF, increases YAP mRNA translation, and that increases cardiomyocyte cell proliferation and leads to cardiac regeneration. It is also identified that M6A plays a major role for myocardial M6A demethylase FTO in regulating cardiac function. There have been different researches and studies going on in epitranscriptomics. The bottlenecks of this field. This field also has technical hurdles. Right now, the methods to map and detect M6A is crude. As mRNAs are abundant, they are hard to identify. That methods require large samples, and they are ineffective at quantifying how M6A levels change over time. The lack of publicly available small molecular inhibitors for studying epitranscriptomics proteins is also another issue. Even if the companies can develop selective METTL3 inhibitors, it's unclear how many people would benefit from them. Well, the leukemia studies by Jeffrey have showed that M6A levels are too high. His leukemia studies showed that the opposite, that the M6A levels are too low. Other studies have suggested the contradictory results. It is crucial to know that epitranscriptomics in different cancer cells. Otherwise, giving the wrong person a wrong METTL3 inhibitor might make worse things. Yes. This field, epitranscriptome. Yes. More epitranscriptomics RNA modifications are yet to be discovered. It needs a very small molecule sampling, but there are no equipments that can be used. Only large equipments are used, like they are used for the genetic modifications. So epitranscriptomics need to be developed, as it is booming in the next decade. Future of pharmacy or the medicine is genetics and genetic modifications. It is applied in cardiac, in cancers like breast cancer, in cardiac diseases. heart diseases like cardiomyocytes, where a regeneration of cell can take place. To observe this high throughput they are used, and for yes. mRNA. I haven't searched. It is still developing. There are only 3 startups up to now, that is in New York and Germany. So it should be developed further. It started the development in 2008. There are only few researchers going on it, as if there are no advanced technologies. I have searched 4 to 5 publications. No, not in India. Thank you.
Hello. Hello. Hello. Mike.
हेलो हेलो हेलो पूरा नजदीक रखना, पूरा नजदीक रखना। यहाँ से बात कर रहा है। हेलो हेलो कौन सी बात? ऐसा नजदीक रख के बात करना है ऐसा। और हेलो
Should I start? Hello, good afternoon, everyone. I'm very glad and excited to be a part of this conference. I'm going to, myself, Hiba Ahmed, and from Deccan School of Pharmacy, from Masters of Pharmacy, Pharmaceutical Analysis. I'll be presenting with my friends, that is Javeria.
Good afternoon, everyone. This is Javeria Fatima from Deccan School of Pharmacy.
Good afternoon, everyone. This is Sabha Konen from Deccan School of Pharmacy.
We are here to present on the major effects, that is microplastics, human exposure and its occurrence. Today, I'll start with presenting this a century, a little bit away from the century. We didn't know what is plastic, and it has been invented, and today we cannot imagine our life without the use of the plastics. And this is the major disaster and the havoc which is causing. What are plastic? Plastic is a heterogeneous mixture with a range of 0.1 to 5,000 micrometer. There are varieties of the plastics, in which the major type is thermoplastic and thermosettings. Majorly, the difference between these two is the thermosettings are the plastics which cannot be remelted, and whereas the thermoplastics are which can be melted and which can be reused, and that is the major warning to the human health. Thermoplastics, in which the major difference is what are the different types of thermoplastics. Thermoplastics are nothing but polyethylene, polypropylene, polyvinyl chloride. These are the major different types of plastics used.
And the major identification techniques used for the identification of microplastics, which is being a disastrous havoc these days, are the various identification methods are physical characterization and chemical characterization. In physical characterization, the various techniques used are transmission electron microscopy and scanning electron microscopy. In chemical characterization, FTIR and the hyphenated techniques like GCMS are being used. The major sources of microplastic, as we know, without plastic, today it has become like we cannot imagine our life. So the major source of plastics is the water itself, because per day a human ingests about a size of pan card size of like a card size of microplastics daily. So water is the major source, and also the various things that are used for packaging, the water bottles, all those, due to various stresses, the plastic microparticles, the macroparticles of the plastic becomes the microparticles, and also the nanoparticles, which are being digested and mainly being absorbed in various organs of our body. Even these are being transmitted to the placental. These are carrying they are crossing the placental barriers also, and also even these are present in the breast milk. So it is a major issue these days.
How we can prevent plastics? Plastics can be prevented to avoid seashell consumption, and also we can avoid using free use of plastics. And hereby we have the conclusion is we can bring the new evidence of weight and the quantitative data in plastics. Thank you.
So what do you want we can suggest that avoid plastics more and more as we can.
Very important. Yes. In that time, so we got that 3 names were given.
Certificate, right?
Good afternoon, everyone. My name is Shravanti. I'm from Geetham University. I'm currently pursuing my final year B Pharmacy. So today, my topic is about hydrotopic solubilization technique. So let's get into introduction. Most of the chemical entities which we use for solubilization techniques are poorly soluble. And the technique which I used is clopidogrel bisulfate drug, and it comes under BCS classification 2, which indicates low soluble and high permeability. So the hydrotropic solubilization is a new, simple, and economic technique. In this technique, we are using clopidogrel bisulfate as a drug. This drug is used as an anticoagulant and platelet inhibitor, so which helps in preventing heart attacks, heart strokes, and also blood circulation disorders. So the main aim is to formulate and evaluate the clopidogrel bisulfate by hydrotropic solubilization technique. In this, we'll know about the low solubility by pursuing different methods. Objective is to formulate hydrotropic of clopidogrel bisulfate for enhancing solubility of the solubility and bioavailability. The materials and methods. The materials we used in this technique is clopidogrel bisulfate and also acacia, sodium benzoate, sodium saccharin, methanol, and also water as usual. And the methods I performed is UV spectroscopic analysis, in vitro dissolution studies, and also FTIR analysis. The technique is hydrotropic solubilization technique. Evaluation parameters are determination of pH and also porability, sedimentation volume, and in vitro dissolution of suspension. And this is the formulation which I used for my clopidogrel bisulfate. I have taken a pure drug and also 5 formulations with different chemicals. Acacia, which is used as a binding agent, and also sodium benzoate, sodium saccharin, and methanol. And results standard calibration curve. We performed standard calibration curve of different dilutions. So this indicates analytical method for hydrotropic solubilization technique. Next. And this is the in vitro dissolution graph. In this F3, which indicates the optimized formulation, and F4, which is a pure drug. The optimized formulation has 98% of the drug is dissolved. So by performing dissolution studies, we can able to know the solubility of drugs. And pH will find about optimum pH environmental conditions, and pourability will know that the drug, it is easily pourable, and sedimentation volume, depth ranges between 0 and 1, in which if F is equal to 1, there is no sedimentation formed. Next. To conclusion, by performing this hydrotropic solubilization technique, we can find drugs are poorly soluble, and these are bioavailable. Thank you.
Still doing FTIR and XID. Yes, ma'am. Within time limit, I did this thing. Yeah, FTIR and XID. Yeah, yes. Yes, ma'am. Yes.
Your name?
Good morning, everyone. So I'm Sukanya Jana, a final year student from Geetham University. So today I'll be presenting about the topic co-crystal, a novel approach to enhance the solubility of valacyclovir hydrochloride. So cold sores, it's a very common thing that we find. So it's basically a painful blisters which appears around our skin in the mouth. So valacyclovir is the choice of treatment which is used for this. So over here, the aim of the study, the aim of the study is that we are preparing fast dissolving tablets of valacyclovir hydrochloride. Next slide. So yes, fast dissolving tablets of valacyclovir hydrochloride by employing co-crystal method. Over here, the method of preparation that we have taken is liquid assisted co-grinding. And then the finalized optimization product is been punched into tablets. Next slide. Okay. So materials that we have used here are written, and the methodology we have followed is basically starting with calibration curve, then followed by formulation of co-crystals, followed by evaluation studies of co-crystals, and then preparation of dissolving tablets using the optimized formulation from valacyclovir hydrochloride, followed by evaluation studies of the formulated tablet. Next slide, please. So for co-crystal formulation, the formula that we have used is there. So we have taken succinic acid as a co-former and the plain drug as valacyclovir hydrochloride. And we have prepared 4 formulations in different ratios, that is 1:1, 1:2, 1:3, and 1:4. From this, the plain drug calibration curve that we have plotted, from there we can see that it is a linear plot indicating with an R-square value of 0.959. And as we can see, there is the dissolution of co-crystal formulation of 4 formulations. And from this, we get like in the dissolution, we have used pH 6.8 phosphate buffer. And in this, we have taken a 40-minute time period with 10 minutes, 20 minutes, 30 minutes, and 40 minutes as its timestamps. And as we can see from the graph that formulation 1 is the best formulation, that is 1:1 co-crystal ratio. Next slide, please. Yes, so as we have got to understand that formulation 1 is the best formulation, so we have went ahead and formulated into tablets and punched them as fast dissolving tablets. And this is the formula that we have followed. And then we have used, after punching the tablets, we have done the evaluation studies. The first one being physical characterization, where we have seen the pH, drug content, weight variation, thickness, hardness, friability, disintegration. And all of these tests were within the acceptance limits. And we have also done, obviously, we will do dissolution of the optimized formulation that is in the tablet formulation. So we have seen a 96% drug release after in the 40-minute mark, proving that the drug has been highly like soluble and giving high bioavailability. Next slide, please. Yeah, so we have went ahead with the FTIR analysis of our plain drug, that is valacyclovir, and the succinic acid And from both of these graphs, we can see in the optimized formulation, it consists both, indicating that the drug and the polymer is highly compatible and there is no such interaction to cause any harm. Next slide. And we have also done XRD graph. So in this, as we can see, the valacyclovir graph has high peaks, whereas there is a shift in peaks in the optimized formulation graph, indicating that the drug has been optimized, like the plain drug has been optimized, and we have gotten a better formulation out of the plain drug. Yes, so in the conclusion that we can see that the fast dissolving tablets using this co-crystal method has been successful, and it can be prepared by incorporating different ratios, but 1:1 is the best formulation. And in addition, evaluation studies were satisfactory and within official limits. And as I said, F1 being the most favorable one. Next slide. Thank you very much to Geetam University, my mentor, Ms. Haima ma'am, and also JNTU for giving me this opportunity to present my work. Thank you. Ma'am, valacyclovir. Ma'am, it's like category 3. Oh, it is an antiviral drug, ma'am. Yes, ma'am, definitely. Ma'am, because as much as I understood, like in my literature review, that it is one of the best best drug used for cold sores. So we have went ahead and chosen, and also because it's the drug that was available in our college. Fast dissolve, super disintegrant, ma'am. So in my case, I have used cross-carbonyl sodium. Thank you, ma'am.
Good evening, one and all. My name is Ankita and I'm from Geetam University, pursuing my final year of B Pharmacy. So today I'm going to talk about formulation and inventory evaluation of amlodipine floating tablets with usage of different polymers. So my drug today is floating tablets. The idea of floating tablets came because the amlodipine drug is usually sold in immediate release. So I thought, why not formulate in sustained release? Sustained release amlodipine bislate, it is a category of calcium channel blockers, which is used for hypertension and angina pectoris treatment. So amlodipine bislate has high solubility in gastric pH. So floating tablets was the best idea for this. The main aim is the present of the project is to evaluate and formulate the amlodipine floating tablets. The objectives are to carry out the extensive literature review of the amlodipine formulation to increase the bioavailability and to have the best optimized formulation. So materials I've used here is amlodipine, the drug, and HPMC, the polymer, and the sodium bicarbonate, citric acid, abyssal magnesium stearate, talc. The citric acid and sodium bicarbonate acts as the effervescent agent in these floating tablets. So here F1 was the optimized formulation for the current project, where all the drugs were evaluated before getting tablet punched in the pre-formulation. So pre-compression parameters were done on the physical mixture of the tablet, where after this was passed, and then the post-compression parameters were done on various aspects like thickness, hardness, fiability, weight variation, continuous uniformity, and floating lag time and total floating lag time as well. The results and discussion were for this, our calibration curve was done where it had a linear, you can see a linear graph, and an inventor dissolution test. It was done for dissolution test, obviously. So here the F1 was proved to be a optimized formulation. So here FTIRs analysis was also done to see the drug and excipient interaction where there was no much interaction. It was all safe to use. And the summary of this and conclusion is, in conclusion, the gastroretentive effervescent floating amlodipine floating tablets may be developed to use in actual clinical practices. The floating tablets were prepared by incorporating different combinations of polymers. Moreover, finding that the pre-compression and post-compression were satisfactory with the results what we found. In addition, F1 was showing the optimized formulation that potentially can be developed and get desired properties of drug release profile. I thank my college for giving me this opportunity to conduct my research studies, and my mentor, Haima ma'am, for this. Thank you. Ma'am, they are not much available because right now we have just immediate release. We need to increase the bioavailability of a drug. So floating tablets can have the sustained release property. No, for now it's not. We are about to prepare. So we need effervescent agent because it has to float on the surface of the HCl. Examples like sodium bicarbonate, citric acid, tartaric acid for the buoyant force to float the tablets on the pH. Because the first formulation, when I tried, it was floating. It had the lag time as soon as possible. It started floating in 35 seconds. It came up and it had sustained release for more than 24 hours. And seeing that, I could go for the next formulations as well. I have done hardness, then thickness, then I have done friability, then I have done for lag time and floating time also I've seen. And then for FTIR analysis also I've done to see the drug and excipient reactions also. Thank you, ma'am. Hello. Donna?
Good afternoon, everyone. Myself, I'm V. Anusha. I'm pursuing my PhD as a part-time research scholar from SRM Institute of Science and Technology, Chennai. So today my topic of presentation is effectiveness of natural constituents from various plant sources like Delanix resia, Terminalia tomentosa, as well as from Trigonella phainum graecum and Abelmoschus esculentus as excipients in formulating solid orals. So what is the role of these natural constituents obtained from the plant sources as an excipient in formulating solid orals? So coming to the introduction, we don't need much introduction about excipients because we all know that excipients are the functional components of any dosage form. It can be a tablet or a capsule or a suspension or a syrup, whatever it is. It may be a solid oral or it may be a liquid, semi-solid, whatever. So excipients play a major role. So based on their origin, these excipients can be like natural, synthetic, semi-synthetic. So my topic of interest is about natural constituents. So why can't we use natural constituents from plant derivatives in formulating various dosages? So coming to the introduction about these plant derivatives, most of the plant derivatives or extracts are mucilages or gums in their origin. So these are polysaccharides in origin. So if we see into the literature, we can see that many of the mucilages and gums extracted from the plants have been used in formulating various extended release tablets, pulsatile dosage forms, enteric tablets as well. So seeing the introduction about mucilage, mucilage is nothing but it is the plant extract which is produced within the cell. So it is an intracellular product. Next.
So coming to the gums are nothing but these are the product which is produced in the plant because of maybe abnormal growth or due to any incision or injury, etc. So on a whole, we can say that these plant derivatives can be used as so and so, like disintegrants, stabilizers, thickening agents, suspending agents, as well release retarding agents, as enteric polymers. So coming to so I chose 4 plants, and within these 2 plants are already established. We have well-versed literature regarding 2 plants. Coming to that, in that first one is Delonix regia. So it is a plant belonging to the family of Fabaceae. So Delonix regia, we have well-established literature regarding this, and the excipients which are obtained from especially from the seeds of the plant. So they have been used as a disintegrant. They have been used as release retarding agent. They have been used as formulating pulsatile dosage forms. If you can see the examples here, the seed gum, it can be used as a binder. So we know that the same excipient will function differently. If you can take starch. So starch at so and so percentage, it can be used as a binder. It can be used as a diluent. It can be used as a disintegrant. In the same way, natural constituents, if you vary their concentration, you can use the same excipient for various functionalities. So you can see, you have even marketed formulations, quatapine fumarate. It has been marketed by using the seed gum of Delonix regia, and it is an extended release preparation. So here I can say that at low concentrations, the seed gum is used as a disintegrant, and at high concentrations, it can be used as a release retarding agent. So we can see the examples which are available in the literature already, the established works. So you can see pulsatile floating system, pulsatile drug delivery of theophylline is also available. So theophylline is a bronchodilator. So mostly we will be seeing nocturnal asthma or early morning asthma in patients. So these pulsatile dosage forms will release the drug after a off-release period. So even such type of dosage forms can be formulated using the Delonix gum. And next, you can see in the literature, you have research work based on formulating diclofenac matrix tablets. So yes, again, at a concentration of 18%. So when the seed gum was used as at a concentration of 18%, the release can be extended up to 12 hours. And we can also see that you can compare with Tragacanth and Acacia, the already established gums. So this Delonix regia gum is no lesser than these gums, and it can be used at a concentration of 1 to 1.5% as a wonderful suspending agent. So next, coming to the next plant, Terminalia tomentosa. So this is, we can say this is under research still. So only few functionalities of the plant derivative have been identified so far. So you can say that it is identified as a binder and as a matrix forming agent. So only 2 functionalities of the plant have been identified still. And in the literature, if you see, going back into the literature work, you can see alprazolam tablets were formulated using this gum obtained from Terminalia plant, and it can produce an extended release up to 14 hours. So you can say that as a natural excipient, the gum obtained from Terminalia plant can be used as a binder and as an extended releasing agent in formulations. So next, coming to the third plant, Trigonella phenum graecum. So we know that this is a plant from the common name of fenugreek. So even with Trigonella, you have well-versed literature is available. So the mucilage which is obtained from fenugreek, it can be used as with many functionalities. It can be used as a film forming agent, as a disintegrant at a different concentration, as a film forming agent, as a floating agent. So all these research has been done, and still it can be used as a very good excipient in formulating various type of dosage forms. So we can see that it is used in formulating even fast dissolving tablets of valsartan. So these are the other few examples. So various literature is available for formulating various dosage forms. So with the nifedipine and carvedilol and propranolol hydrochloride, it is used as an extended releasing agent. And even fenugreek mucilage is used in mucoadhesive dosage forms, as well as it can be used in nasal preparations, etc. And the last one is with Abelmoschus esculentum. And nowadays we can see many dosage forms have been formulated using ochra mucilage. So it is known with the common name ochra, we know that. So the main component again here is the mucilage. So you can see here, it can be used as a binder, super disintegrant as well. So at a concentration of 4%, it is used as a super disintegrant in formulating various dosages. Next. So we can see some other examples where it is used as an extended releasing agent in formulating pentoxypyrin tablets, lamivudine tablets as well. Conclusion. Next slide. Yes. So finally, in the conclusion, I can say that as these natural excipients are like compatible, degradable, and they are with very less side effects, they are beneficial to the patients, as well as they are renewable. That is, we cannot say it is extinct. So it is very much beneficial to the industries also to find the polymer. You can prepare polymer by chemically modifying these excipients again. So it is beneficial to the patients as well as to the industry for formulating new dosage forms. Thank you. No, ma'am. It's actually, I'm planning for my PhD work actually. So on natural excipients. Yes, ma'am. Yes. So still it's under like planning to use which gum and which excipient. No, ma'am. Actually, I'm thinking of okra. Yes. So I want to give a review on that. So that is the reason I presented this. Thank you. Any? Polymer. So if we, yes, synthetic polymers, we can say something like which are not degradable. Synthetic in the sense something like PVA or PEG, something like, suppose we can use PVA. It is as an ingredient, as a binder or something. But if we use a mucilage, what kind of toxicity in the sense? Maybe as of now, I'm talking about the degradability. It may not be degradable or it may show some side effects. Yes. Yeah, true. First thing is we will do the FTIR and pre-formulation studies. There itself, we will understand what is happening between the excipients and between the drug. So as of now, based on the literature which is available with natural excipients, the studies indicate that they are very much compatible. So all the FTR graphs or this, what do you say, TGA, DSC, whatever. So in every you have all those studies. So it indicates that they are very much compatible rather than interactive. यहाँ पे दबाना.
Hello, good afternoon, everyone. Myself Prabhu Shreya Ajay from KLE College of Pharmacy, Hubli. I'm an assistant professor. Title of my presentation is Preparation and Characterization of Different Polymorphs and Formulation of Immediate Release Tablet of Maclofin Hydrochloride. Introduction. Polymorphs is ability of a drug to exist as two or more crystalline phases that have different arrangements and or conformations of molecules in crystal lattice. When polymers and or solvents of pharmaceutical solids can have different chemical and physical properties that impacts on processability and quality of drug. Selecting polymer exhibiting greater solubility and bioavailability is important in pre-formulation studies. Okay. Okay. Aims and objective. The aim of a present work is to improve solubility and dissolution of Maclofin hydrochloride by preparing polymer and formulate into an immediate release tablet for better onset of action and patient compliance. Then materials and method. The supersaturation, it was prepared by different methods such as saturation method, supersaturation method, photostability method. Supersaturation method gave the best results with ethanol is to water 2:1 ratio at 60 degrees Celsius and crystallization at 20 degrees Celsius, drying at 100 degrees Celsius for 60 minutes. Polymers were characterized for DSC, XID, and dissolution. The formulation table of after preparing polymers was prepared with metastable polymer 250 mg of McLaughlin hydrochloride, microcrystalline cellulose, then super disintegrant sodium starch glycolate, talc, magnesium stearate, and aerosol with specific quantities. The metastable polymer and super disintegrant was mixed, and to this microcrystalline cellulose were added, and it was passed through sieves and blended with aerosols, purified talc and magnesium stearate, and the tablets were compressed. Results and discussion. The DSC graph of McLaughlin hydrochloride gives a crystalline peak at 274.32 degrees Celsius, and the in supersaturation method it gives a amorphous peak at 260 degrees, and also the other the exothermic peak at 264 and 291. The XID also shows the crystallinity of the graphs, and it shows different peaks. Then the dissolution of McLaughlin hydrochloride as a pure drug and the polymer is done and compared. It releases at very less time, when we make polymorphs, it releases in very less time, and the polymorphs are successful, and even the after the tablet compression, the polymorphs also gives a better tablets. The pre-compression parameters were done such as bulk density, tap density, and Hausner's ratio for angle of repose and compressibility for the tablet before the compression of tablets, and after compression, weight uniformity, hardness, friability, disintegration, and wetting time, drug content, and water absorption was performed. The wetting time summary. Among all the formulation, metastable polymer prepared by supersaturation method using ethanol is to water 2:1 as solvent shows drug release of 93.13 and in 40 minutes, and immediate release tablets which were formed with this metastable polymers with 5% of super disintegrant, that was sodium starch glycolate, showed minimum disintegration time, which will give better patient compliance. And the drug release was 98.71 in 40 minutes. And conclusion, metastable polymer of McLaughlin hydrochloride prepared by supersaturation method in ethanol is to water 2:1 shows better solubility and resolution rather than prepared other polymers which were prepared by saturation method and photostability method, and it gives a better immediate release formulation and a better drug release.
Okay, you said you metastable polymorphs of McLaughlin. Yes. And you have used the super disintegrant.
Super disintegrant.
Okay. There is a super disintegrant whenever you use, there is a possibility of drug release is more.
Yes.
So what is your novelty in your presentation?
Ma'am, McLaughlin hydrochloride had release after like almost 4-5 hours. So I have increased its release by making it as a polymorphs first preparing polymers by supersaturation method and then making it into a using super disintegrant and preparing a tablet.
Okay. So when you release the more mefloquine,
yes,
you know that mefloquine is going to deposit in the liver.
Yes.
Isn't it?
Yes.
So don't you think the drug toxicity will indicate if you release more drug?
Yeah.
Then what is the novelty of your work actually? By doing a super disintegrant and increase the release, which might end up in toxicity.
So, ma'am, actually the toxicity studies
You have to do.
Yes.
Otherwise,
yeah.
Okay. Okay. Any questions? Okay. Thank you.
Thank you.
Your certificate will give you in a minute. Okay, please. Next participant.
Good afternoon, everyone. Myself, Dheeraj Krishna. I am doing M.Pharmacy in JSS College of Pharmacy. Today, my presentation topic is on US FDA safety regulations on the radiation emitting medical devices. Not only the US FDA not only releasing the guidelines for the these medical devices and also for the different electronic devices which releases the radiations. In that, I took the medical devices. When we talk about the medical devices, X-rays or the missions which releases more of the radiation. In the scanning, while taking the scanning, the radiation levels will be releasing more on the body. By the way of the scanning, the internal organs will absorb the radiation and the remaining will be emitted. That the result will be shown to the outside. That result will be evaluated. There will be different types of the scanning methods. Some will be taking only single snapshot. Some will be taking different continuous images. Some will be taking overall body. Like that, in that, radioscopy and also the fluoroscopy and other CT scans. These are the different. These are the different types that the scanning will be done. When we talk about the radiography, there will be the only snapshot, the only one snapshot can be taken. When come to the fluoroscopy, the different imagings can be taken continuously. And also in the CT scan, whole the body will be gone through to the radiation. In the aims and objectives and under the guidelines, the US FDA releases the information under the guidelines to the patients and to the doctors and also to the scanning people who are doing scanning. And also these are the steps that will make the patients safe who are going through the scanning. The materials and methods. I collected the matter from the published articles and also from the US FDA main regulatory website and also CFR, that is a Code of Federal Regulations. Under the results and discussion, the results will be the discussion to the doctors. According to the US FDA, it gives information to the doctors that they should be taking some guidance and also the while pursuing the a medical device, they should be taking care of the medical device, how much the radiation is emitting the medical device. And also information to the imaging scanning team. The information from the US FDA is they should wear the proper equipment, and also dressing should be different. They should not go with the normal dressing while taking scanning. Well, come to the hospital administration. Even hospital administration also have the role in maintaining proper health of the patients who are undergoing to the scanning. There we should be the proper trained a person should be there for taking of the scanning. When come to the under the guidelines, the US FDA also give regulations to the if anyone observes the adverse drug reactions or any side effects after the radiation, that should be informed immediately to the regulatory agency through the MedWatch. The MedWatch is the platform where the any person can report the adverse reaction to the agency so that the agency can take any action on that. There will be the different methods of the submitting of the adverse reactions. In that, some will three types are the preparing the electronic copy and submitting in the submitting to the portal, and another is preparing the electronic copy and submitting through email. Another one is the preparing normal Word document and submitting through the mail. When come to the and also USFDA also give the regulations that how the medical device can enter into the market, like what is the steps that any manufacturer should take care to keep the medical device into the market. In that first step, they should be look for the local agent, so that the local agent will take the whole responsibility in releasing the market, releasing the medical device. Second step is the listing. In the manufacturer should give the total list of the medical devices which he want to release into the market in the US. And also third is the filing. In the filing, there should be given all the authorizations, GMP certificates, and all the what are the authorizations they have. When come to the import terms, it is not like all the people can import the medical device in the US. There will be just certain regulations for importing the medical devices. They should give all the specifications, what are the tests they did. They should give the evidence that it will not show that much radiation effects. It will not cause cancer. When come to the conclusion, not only the USFDA, not only giving regulations and sitting silently, it is not like that USFDA is going with. USFDA is collaborating with the different companies, and also have they have the different collaborations with the companies, like they have notified bodies to give the certificate for that medical device, so that it is a proof that there is no more adverse effects than the benefits which are really coming from the medical device. I thank my college, JSS College of Pharmacy, Mysore.
Good. Okay. But you are given in medical devices, isn't
Yes,
it?
ma'am. Yes,
This
ma'am.
is a review.
Yes, ma'am.
Okay. So are you able to use any of these medical devices which you have presented today?
Sorry, ma'am.
Are you able to? I mean, did you gone through any training?
Oh, no, ma'am. No, ma'am. I am from regulatory regulatory affairs, ma'am. I'm pharmacy in regulatory affairs.
Regulatory affairs. Okay.
Yes.
All the best.
Thank you, ma'am.
We'll give you the certificate in a minute. Okay.
Go ahead. Go ahead. Go ahead.
Participant. Next participant. పేరు రాసుకుంటున్నాను. क्या हुआ? You can start.
Good afternoon, ma'am. So good afternoon. So my topic is advancement in solubility enhancement techniques. So we all know that solubility is the main aspect of formulation when we formulate a drug or medicine. So according to BCS class, there are it is classified into 4 classes. So the class 2 and class 4 drugs have the low solubility. So enhancing the solubility, there are many conventional techniques like solvent evaporation, change in pH, temperature and all. But now as the number of drug candidates are increasing, so the more lipophilic drugs are increasing. So they have poor aqueous solubility. So for enhancing their solubility, we have introduced new novel techniques, advanced techniques. So here I have discussed 6 of the novel techniques. So the first technique is liquid solid technique. So in this, basically the liquid solution of the drug is converted directly into free flowing powder, so that the compression of the tablet is easy. The next technique is the co-crystallization technique, in which the API and the excipients are crystallized or co-crystallized in the form of salt, solvate, hydrates, or they are converted into polymorphs. And the third technique is spherical agglomerization, in which crystallization and agglomerization are combined, and small sphericals are prepared, in which in this technique there are actually 3 types of solvent used. In first solvent, the drug is dissolved. The second solvent, drug is partially dissolved. When these 2 solvents are mixed, a crystal is formed. To convert these crystals into agglomerations, there are third solvent is added, which is called as bridge liquid or bridge solvent. When we add this bridge liquid, the small spherical agglomerations are formed. The fourth technique is melt sono crystallization technique, in which the crystallization and ultrasonification techniques are used, combined in both, in which first of all, the drug is heated up to 193 degrees centigrade to make into molten form. And this molten form is directly added to deionized water, which is around 50 to 60 degree maintained temperature. And after addition of the molten drug into the deionized water, it is subjected to ultrasonification. So this for 15 to 20 minutes, then small spherical drug particles are formed, which has a more solubility when compared to the initial form of the drug. The next 2 approaches are one is the prodrug approach and one is nanotechnology approach. As we know, a prodrug is an inactive form of a parent drug. When introduced in the body, enzymatic or chemical changes, it forms gets its parent form. So this prodrug approach has shown nearly from hundreds to 1,000 times of increase in the solubility. And the next last approach is the nanotechnology approach, which is generally majorly used for the potent drugs, for increasing their solubility. In nanotechnology, there are various techniques like SMEDS, like self-emulsifying drug delivery system, and SNEDS, self-nano-emulsifying drug delivery system. And others are dendrimides and more of like more nanotechnology techniques, like we use so the SNEDs and SMEDs are the main important techniques here, along with the nano suspensions, nano emulsifications. So here I conclude that there are these approaches have to be increased because the number of drug candidates which are which has poor aqueous solubility basically. So as the pharma sector is increasing, new drug candidates are discovered, so the solubility problem stays. So so reducing the solubility problem, more and more of these novel technologies has to be increased. Now we have seen, like initially crystallization was a different technique, this sonification was a different technique. So melt like using temperature, we dissolve we enhance the solubility. So here these novel techniques have brought together 2 to 3 techniques and made a novel or advanced technique. So such more work can be done on this sector and aspect for increasing the solubility. Thank you.
Okay, so solubility enhancement techniques you have.
Yes.
did. So you are doing M-Pharmacy?
Yes, ma'am. I'm from first.
Okay, have you doing any project on that?
Not yet, ma'am. The second year I am planning for this one of the technique I'll be planning for the project.
Okay, which technique you are planning now?
Ma'am, I'm interested in this melt sonocrystallization. It's an interesting technique. So either that or co-crystallization technique.
Okay. What is the difference between co-crystallization and melt crystallization techniques?
So ma'am, in co-crystallization, what happens? There are the two. One is the excipient and the API. We like we combine them in a different form. The like amorphous form is generally converted to polymorphous form. Different, its form is changed. So for example, there's amorphous form. When we apply this co-crystallization technique, we can change it into salt form, solvate forms, hydrate forms, and otherwise this amorphous form is break down into little like little agglomerates to form a polymorphs. So in melt crystallization, it's that the drug is melted. So melted and it's subjected to sonification. So because of sonification, the particle size reduces and it gets a spherical shape.
Okay. Okay. All the best.
Thank you. Thank you. Soham. Ji Pulare.
Okay. Ayupoyinda? Next. We'll give it here so we can take a picture.
Good afternoon to one and all present here. So my name is Aisha Mohammed, currently pursuing MPharm in JNTUH, and I'm here to speak on current advancements in immunotherapy. Yeah, so basically until recent years, the immunotherapy basically evolved around surgeries, chemotherapies, radiations. Until 2011, FDA approved a drug named ipilimumab that was used for the treatment of metastatic melanoma. So that time, this drug actually led to this treatment called immunotherapy. So immunotherapy became the fourth leg in the treatment stool of cancer. So basically immunotherapy is using our own immune system to treat against diseases and cancer. So how does immunotherapy works? It basically works by recognizing the aberrant cells. Aberrant cells are as in those which are different from the normal cells. They recognize those, eliminate, slows and stops the growth of malignancies. It can be either given in the form of IVs, that is intravenous directly into the veins, intravesicular directly into the bladder, topical like as in creams, and oral in the form of pills or capsules. So the main aim of immunotherapy is to strengthen the body's immune system so that it can eliminate tumor cells and produce long-lasting anti-tumor immune response. And the objectives are development of novel immunotherapeutic strategies. That is, researchers are constantly exploring new methods to use our own immune system to fight against the diseases and the autoimmune diseases. And also they either by making new methodologies or by improving the existing ones. Coming to the improved patient outcome, immunotherapy has shown promising approach when it comes to the treatment of cancer. It has been treating nearly 15 types of cancer, including lung, bladder, and kidney, also melanomas. So overcoming resistance to immunotherapy, this is one of the biggest challenge which researchers are again undergoing work. For example, identifying the biomarkers that can predict resistance. Developing combinational therapies as in using immunotherapeutic techniques among each other or by using it with traditional treatments like chemo or radiotherapies, which is basically proven to be more effective than that of the individual treatments. Then developing safer and more effective immunotherapeutic agents. So when compared to the traditional treatments like chemotherapy and radiations, immunotherapy has proven to be more effective and having less side effects. Personalized treatment, this is basically the trending thing that researchers are basically going on. It has the potential to be personalized based upon unique individual, immune system. So the current researchers that are going in immunotherapy are finding solutions for resistance by using combinational therapies, finding ways to predict responses because not all patients respond to it. So researchers are ongoing researches for this one, and learning more about how cancer cells suppress immune response against them. Also, even though immune therapy is having low side effects, it still contains some side effects. So in order to reduce those side effects, researches are going on for the treatment of immunotherapy. So the methodologies that has been used are CAR T cell therapy, immune checkpoint inhibitor, cancer vaccines, combinational therapies. So CAR T cell chimeric antigen receptor therapy, here it enhances the ability of the T cells present in the immune system itself. So the WBCs are taken from the patient suffering from tumor. It is modified or altered or modified. Then it is synthesized, grown in labs, and put back into the patient's body via IV. And coming to the immune checkpoint inhibitor, it basically blocks the immune checkpoint. Immune checkpoints are the normal part of our immune system that keeps immune system from being strong. So drugs, such drugs allow the immune checkpoint to be blocked to show effect against the cancer. These immune checkpoint basically acts like switches that should be turned on and off for a response. So the immune checkpoints are PD-1, PD-L1, and CTLA-4. So PD-L1 inhibitors, nivolumab and pembrolizumab. So the first drug that came for immunotherapy was ipilimumab, which is used for metastatic melanoma. That is the CTLA-4 inhibitor, one of the immune checkpoint inhibitor. Coming to the cancer vaccines which basically boosts the immune system. So the first drug that was there, this is one of the dendritic vaccines. Vaccines can be either cancer vaccines given in the form of DNA vaccines, peptide vaccines, or dendritic vaccines. They're made in basically 3 ways: using our own immune cells, or by tumor-associated antigens, or by using our own dendritic cells. Dendritic cells example is suppressor T. Coming to the combinational therapy, which have shown more effect when compared to the individual therapies, combining immunotherapy with one another, that is with immune checkpoint with T-cell therapy or the cancer vaccines, or else using combinational therapies along with that of the traditional therapies, which enhances the efficacy as well as diminishes the toxic effect. So coming to the result, immunotherapy has actually shown promising effect. And the average rate response for treatment is nearly 20 to 50%. Also, the treatment basically depends upon the type of cancer and the immune system of the patient as well. Then almost 40% of the people nearly suffer from cancer, out of which it has been shown that 15.5 million has been survived cancer until 2016, and which is expected to surpass 20 million by the year of 2026. And the drug ipilimumab was approved in clinical trial phase 3, which showed 24% of survival rate up to 2 years. For some patient, even showed 4.5 years. Yeah, it's done almost. So this drug named pembrolizumab, this also showed 5 years of survival rate. Its response rate changed from 5.5 to 15% for non-small cell lung cancer. And for prostate cancer in 2009, 1100 clinical trials were gone, out of which 12% of the techniques were immunotherapy. Next. Okay, so coming to the again first lung for melanomas, like the blood cancer type, ipilimumab showed 21% response rate for 3 years. And for nivolumab and pembrolizumab, it showed an increased rate. That is a modified version of ipilimumab. These are the monoclonal antibodies. It showed 34% rate at 5 years and 44% rate at 4 years. And combination of interleukin 2 and CTLA-4 blockades showed prolonged survival rates and reduced tumor. Next. So as you can see in the slide, this the blue line is the that's the conventional chemotherapy. When you compare all of these, the second one is first generation immunotherapy, second generation, and last is the combinational therapy. So as you can compare how the survival rates are increasing. So the combinational therapies have been shown to have more effective than that of the conventional therapies. Next. Yeah, coming to the summary and conclusion. To summarize with, precision medicine is at the heart of immunotherapy, one of the hottest area of cancer therapy. Immunotherapy, specifically combinational therapies, have been showing much more effect than that of the individual treatments. And the drug ipilimumab, for example, ipilimumab, that is the immune checkpoint inhibitor, along with the suppressor T, that is the cancer vaccine, both in combination have shown a promising effect. And yes, despite there is a drawback that not all patients respond to this treatment. So researchers are ongoing the reason for why it is happening. And yes. Thank you.
Okay. So like immunotherapy is a precision medicine.
Yes.
Isn't it? So how this precision medicine will be preparing and depending on what?
Depending upon the patient immune system. Like precision medicine is basically how you say, if whether the patient is responding to the medicine or no. The patient, it depends upon all the patients, what do you say, the properties or,
Gene.
Yeah.
We need to talk about the gene. Whenever it comes in the precision medicine, you have to talk about the
Gene,
gene sequence.
Yes, right.
Okay. Okay.
I see. I see.
Okay. Next. One more lady was there. One more lady was there. She did not take.
My poster. Warm evening to one and all gathered here. Myself, Hemaleta. I'm pursuing B Pharmacy in 7th semester in Sri Venkateswara College of Pharmacy, Puducherry. And my poster presentation is about the implication of artificial intelligence in field of pharmaceutical technology in a perspective sense. And I take this as an overview. Okay, when we talk about an artificial intelligence is nothing but an intelligence that mimics or imitates one's intelligence that completely a human intelligence. In recent years, our not only our economy and all over the world economy were facing drastic to mild issues regarding their development of their own field due to either COVID pandemic, or due to wars, etc. So, pharmaceutical industry were not serves as the exception. So, pharma sectors have undertaken various technologies, including artificial intelligence, CRISPR technology, and bioprinting, etc. Among them, artificial intelligence plays an extensive role in development of the pharmaceutical sectors. Then the aim of my presentation is to analyze the upgrades in a perspective sense in artificial intelligence. An objective of my study is to make the prospective generation acquaintance to the knowledge of the artificial intelligence. Then working of the artificial intelligence, a simple working, a basic working I can give. Artificial intelligence is nothing but a computer programmed system that can works with the these lot of algorithms. They can works by deep learning and knowledge perception and etc. They include speech day-to-day life. We are using the artificial intelligence in each and every sectors and each and every softwares. The chief learning is the paradigm of machine learning. The machine learning includes artificial neural networks and etc. Then the roles of artificial intelligence in pharmaceutical sectors. The role of artificial intelligence includes various sectors such as industry, drug discovery, pharmaceutical marketing, patient healthcare, and assisting patient health. Among them, the three sectors plays an important role, such as drug discovery, pharmaceutical marketing, and pharmaceutical manufacturing. First, let us see about the pharmaceutical drug discovery. As we all know, a drug discovery takes about 10 to 15 years for a new drug, from the lead drug discovery to the regulatory approval. So artificial intelligence can make it more effective in by using less capital, using less resource, and decrease the time consumption and etc. Then when we see about the manufacturing, artificial intelligence plays an extensive role by making the continuous production, because India is considered as one of the pharmacy of the world. So continuous production of the pharmaceutical things can make the world better and India's marketing too better. Then we come about pharmaceutical marketing. AI helps in pharmaceutical marketing by predicting the demand and sales forecasting, and also in pharmacovigilance. Then my summary is, humans are the most sophisticated machines that can ever made. So human brain is always thinking about something to make it better than it. So artificial intelligence serves as one of the best technology that human can make. According to Stephen Hawking, artificial intelligence is either a worst or a best thing that will happen in future. So when we talk about the artificial intelligence, it has both the advantages and disadvantages. When we talk about the advantages, it provides lot of boost and lot of development in our marketing. That's it. Thank you. What is your name?
How many presentations there? 10 presentations.
Good evening, everyone. I am Saranya from B.Pharm final year at Sri Venkateshwara College of Pharmacy, Pondicherry. Sri Venkateshwara College of Pharmacy. And my study under differential diagnosis of the Parkinsonian disorders using 11C CFT PET imaging as in silico method, which is a computer-aided drug design. And we'll start from introduction. Parkinsonian disorder is a differential of characterized by tremor, bradycardia, and bradykinesia, rigidity, and the postural of the poor rigidity of the stability, and many characterized by the Parkinsonian disorders is under PST and MSA, which is full formed by the multi-atrophy drug under the Parkinsonian disorders. And the need for the study is under MSA, which is multi-system atrophy, and PSP, which is progressive supranuclear palsy. And it is material which is defined under MRI scan, which can be diagnosed by the Parkinsonian disorders. And this method may be differentiated as 3 categories, which is Parkinsonian syndromes, or always with the neurological disorders that comes under the PDS. And the tables which have taken as a differential parameters with of categories and then a strategy with the percentage materials, that is of 4 types. And the next differential characteristic of regions and the diagnosis of the patient by testing their disease under the organs with the substantia nigra in the presence of the brain. And third, which is taken as a strategy and the characteristics of the region and characterized by a particular person with of the diagnosis of Parkinsonian disorders. And under the summary and the conclusion, to investigate and the diagnosis of the Parkinsonian disorders under a imaging of in silico method. Thank you, ma'am.
JNTU has 2 names.
Good evening, ladies and gentlemen. My name is Sanya Thakur, and I'm from Narsi Munji Institute of Management Studies. With me, I have my co-author Anandita Bagh. We are here to present on the topic National Education Policy and its impact in pharmaceutical industry. So the NEP came into being in 1964 when the government of India constituted a Kothari Commission, which gave its reports and records. And further, the then Honorable Prime Minister, Indira Gandhi, she came up with this idea of the National Education Policy. Further, in 1986, the then Honorable Prime Minister Rajiv Gandhi implemented a new NEP which focused on special emphasis on equal education for all, disparities, women and reserved categories. And finally, in 2017, the Ministry of Human Resource Development developed another final lag of the NEP, which was implemented in 2020.
Coming to the need of our study, while we were conducting this research, we had two main objectives. One was to determine the awareness of students and pharmaceutical professionals about the national education policy. And the second one was to determine their opinion about the impact that this policy will have on the pharmaceutical industry. Some of the major reforms which concern the pharmaceutical industry are the increase in the number of colleges and universities which impart pharmaceutical education, and the various encouragement of internship opportunities and the vocational training programs which will be given to them, the encouragement of impartition of digital platforms so that the students can acquire high quality knowledge from and also acquire online education prospects.
Now, moving on to how we conducted our study, we basically conducted our study through an online platform where we had two sets of participants. The first being the student volunteers. They were asked a question. The question was, in their opinion, which reform do they think is the best for the pharmaceutical industry? The options that were given to them were emphasis on internships, support for entrepreneurial ventures, increase in number of pharmaceutical colleges in the country, and majority of the students felt that giving a support for the increase in internships would be the most beneficial for them. Moving on, we had another set of experiments that we sent across to industry experts. About 50 participants were included in this. We asked them whether they thought that the new NEP, which will single out and phase out the single phase skilled colleges, disciplinary approach by 2040, which is the target of the NEP, will it affect the specialization in the pharmaceutical industry, which is key, as we all know. Most of them agree, that is about 60% of them agree that no such thing will happen in the near future and specialization won't be hurt.
So finally, it can be said that the national education policy is comprehensive. It focuses on early childhood education. It takes into account a holistic approach towards education, and it also promotes inclusivity. As per our survey, we come to an understanding that the students, though are aware of the national education policy in general, they do not have a comprehensive in-depth knowledge about the specific reforms which this policy brings in. Also, moreover, it can be said that the students emphasize more on the internship aspects, which apparently seems more beneficial and tangible to them. Coming to the impact of the policy on the industry, it can be said that this will only be known if about how its implementation will be on the industry, and how much the stakeholders, to what extent the stakeholders adopt this. The stakeholders include the pharmaceutical sector and the educational sector. Also, the industrialists feel that there won't be any much effect on the specialization course as one would have anticipated it to have.
We conclude by saying that the NEP will bring better reforms overall, as it focuses more on practical knowledge than theoretical, which is more important in the pharmaceutical industry. Thank you.
Okay, in national education policy, okay, in 2020, what is the major impact on the students?
Like I mentioned, ma'am, that the NEP 2020 focuses more on hands-on learning and practical experiences than rote learning and theoretical knowledge. So students will be able to choose from a variety of subjects of their interest, and they will understand each and every subject in depth, and they will when you have an interest in a particular subject, you tend to excel in it. And this will help in increasing benefits in every kind of sector, especially the pharmaceuticals, because interest is very necessary here.
Okay, in pharmacy, we have not implemented NEP yet, but the NEP does multiple exit and multiple entries to the any kind of course. When multiple exits that you finish after 2 years, you want to exit, then you'll get a diploma. If you exit after 3 years, you get a degree, or you get a next level of the certificate. For the 2 years certificate, third year, you'll get the diploma, fourth year, you'll get the degree. So like that, multiple entries and multiple exits are available. So you can work 1 year after 2 years of your education, and you can work somewhere and come back and rejoin and complete the your education also. That is the major impact for the students. So if they want to have work experience during your studies, you can do that. That is the NEP highlights of that. Okay. Okay. Next. Co-author and author. The co-author certificates. Author certificate.
Very pleasant. Good evening to one and all who all are present here. Coming to my research title is about formulation and in vitro evaluation of extended release tablet of ketorolac with Eudragit adjuvant. Coming to introduction, most convenient oral drug products such as tablets and capsules are formulated to release the active drug after oral administration to obtain rapid and systemic drug absorption. Extended release of oral drug have better therapeutic efficacy by maintaining steady drug plasma concentration. Eudragit polymer have been demonstrated for its extended drug release, and Eudragit polymer is a cost-effective and broad regulatory, which is also act as a hydrophilic hydrophilic gel forming agent for hydrophilic water forming agent for extended release of tablet. So why we have to need for study is we have the ketorolac polymer is used to extend and L100 are used. It shows 99.71%, 71% bioavailability, which is a good extended release, and which have 10 hours duration of action. We can take once daily administration of the tablet. I'm thankful to KVK College of Pharmacy for providing to complete this entitled work.
Good evening, everyone. I'm K. Shamsundar. I'm a B.Pharm secondary student at Vishnu Institute of Pharmaceutical Education and Research. Today I'm here to present you a topic named Robotics and Automation in Pharma Manufacturing. Next slide. The contents of my presentation are introduction, benefits of robotics and automation, examples of robotics and automation in pharma manufacturing, challenges of implementing robotics and automation, future of robotics and automation in pharma manufacturing, conclusion and references. Introduction. The pharmacy industry is one of the most sophisticated industries which need high regulatory bodies and also a view on the patient safety. With the usage of robotics, we can get high efficient, accurate and more safer drugs. Next slide. Next slide. The benefits of robotics and automation in the pharma manufacturing are it can improve efficiency, accuracy, and also safety. For example, if you use robotics in place of human beings for dangerous activities like with experiments of radioactivity, we would have saved the great scientist Marie Curie. In such conditions, robots are useful. Next slide. The examples of below two figures, those are the quality control missions. If you observe the above two robots, those are used for the packaging of the drugs. Okay. The future of robotics and automation in pharma industries. The use of robotics has been increased drastically in the last few years with the collaboration of new technologies like artificial intelligence and machine learning. There will be a drastic increase in the manufacturing of the pharmaceutical products. Okay. Conclusion. Introducing robotics is very difficult because of the high investment, etc. But by doing so, we can see a bright future in pharma field. These are the references I have taken from. Thank you, sir.
Very good evening, everyone. I am Himabindu, pursuing my BPharm second year from Vishnu Institute of Pharmaceutical Education and Research. Before going to my topic, nowadays, irrespective of age, many people are suffering from cancers. So we have to go for the new inventions to treat other than the traditional methods. So I selected one of those method, that is multi-drug nanoparticles in cancer treatment. Next. These are the contents of the following slides. Next slide. So nanoparticle. Nanoparticles are materials their overall dimensions are like nanoscale under 100 nanometers. These are the solid colloidal particles. Generally, their size ranges from 10 nanometers to under 1000 nanometers. But when it comes to nanomedical applications, the preferable size of these nanoparticles is less than 200 nanometers. And when multi-drug nanoparticles are also a type of nanoparticles which are designed to deliver more than a single drug to the targeted site. Next slide. And multi-drug nanoparticle in the treatment of cancer, in treating cancer, combination of the drugs is more efficient, shows the more efficient way than using a single drug. So by this, MIT scientists have prepared, they designed a bottle brush shaped nanoparticle in which we can load more than single drug, and the ratios can be controlled easily. And the in their study, in a study of mice, they have observed the tumors have shrunk more faster than using a unthreatened same drugs but unthreatened way. Next slide. And control ratio. Using these nanoparticle to deliver cancer drugs allows drugs to accumulate in the tumor site, and they'll reduce the toxic side effects. And because these nanoparticles protect these nanoparticles protect the drug to release, they protect releasing before reaching the target site. And however, in now present, more than only a handful of nanoparticles have get this FDA approval, but only one of those nanoparticles are having more than a single drug. If we want to make a bottle brush that has 2 drugs or 3 drugs or any number of drugs in it, we simply need to synthesize those different drugs, drug conjugate monomers, mix them together and polymerize them. The resulting bottle brushes have exactly the same size and shape as the bottle brush that only has 1 drug, but now they have a distribution of 2, 3, or however many drugs you want within them, says Johnson. This is the words I got from the article. And this image is posted by the MIT scientists. These are some nanotherapeutic methods used in treatment of cancer. Next. So by this, I would like to conclude these multi-drug nanoparticles have emerged a promising approach in treating cancer. But like they are offering advantages over our traditional methods, and as well as their improved drug efficiency and reduced toxicity, and also targeted drug delivery. Despite some challenges, these multi-drug nanoparticles will treatment, but like they treats cancer promisingly.
Good evening. Oh, sorry. Good evening. Myself, Kusmita, pursuing my B.Pharmacy first year in Vishnu Institute of Pharmaceutical Education and Research. Today, my topic for presentation is formulation and evaluation of herbal gel containing allium cepa extract. The objective of this research is to formulate a herbal gel containing allium cepa extract and to evaluate that herbal gel. Gels are the newer dosage forms which are created by entrapment of aqueous hydroalcoholic liquids and colloidal solid particles, which contain aluminum salts and other organic substances. The concentration of gelling agents in gels is usually 0.5% to 2%. External application of gels have certain advantages, like the drug can easily be reached to the site of action. Coming to before. Many people are preferring herbal products these days, one of which is onion. Onion belongs to family Liliaceae. It is a highly valued food and medicinal plant. It contains 89% of water and 1.5% of proteins and vitamins like B1, B2, and C, along with potassium and selenium. Onion has a potential in treating cardiovascular diseases, stomach cancer. Coming to the important materials that are used in the formulation of herbal gel: allium cepa extract, methyl paraben, triethanolamine, carbapol 934, and propylene glycol. Method: there are 3 steps in formulation of herbal gel.
pre-formulation, formulation, and evaluation. In pre-formulation, the characteristics of allium cepa, it has yellow color, odorless, and it is a solid state. In pre-formulation, we have drug excipient compatibility study by FTIR. The FTIR is carried out by drug and formulation. It is used to establish the unknown compound and an analysis for functional group. Next, calibration curve of allium cepa. A stock solution of standard drug was prepared by using phosphate buffer of pH 7.4. This with a wavelength of 257 nanometer using quartz cuvette in UV visible spectrometer. Coming to the formulation, in 50 ml of distilled water, carbapol 934 is dispersed in 250 ml of water, and it is continuously stirred using a mechanical stirrer at a high speed, and it is kept aside to swell, and then this solution is used as the base for the gel. Next. And 5 ml of water, required amount of methyl paraben is dissolved under water bath, and the solution is cooled. The next. The solution is cooled and required quantity of propylene glycol is added. Next. Then further, the required quantity of allium cepa extract is added, and the solution is made to 100 ml using the remaining distilled water. All the ingredients are mixed continuously, and required quantity of triethanolamine is added dropwise to the solution. It is added to adjust the skin pH and give the texture to the gel. The gel is the formulation is filled in the container, and the last of the conclusion. The conclusion is that after these days, people are preferring herbal products and herbal medicinal products. But after the formulation of this allium cepa extract, the F3 formulation found to be good at evaluating points such as homogeneity, pH, and physical appearance, etc. And we found that this allium cepa extract can be used to prepare gels. Thank you.
Well, how many of us have actually ever heard the prompt, Alexa, okay, Google, why not just try one such thing real quick? Okay, here's the prompt. Hey Siri, what's the sunset time today?
Sunset will be at 6:26 p.m. today.
Well, I asked for sunset time to Siri. It said it's going to be 6:26 in Hyderabad today. So precisely, I just got what I wanted or what I asked for right at the tips of my fingers. And speaking of which, this is the era, this is the generation, this is the competitiveness that we are living in. So this is all about, this is what Industrial Revolution 4.0 is all about. A very warm welcome and evening to everyone present here. I, Savan Nagaram from the Narsimonji Institute of Management Studies, deem it an honor to stand before you and present my poster titled Digital Transformation of the Pharmaceutical Industry, Harnessing the Power of Industry 4. Now talking about Industry 4, Industry 4.0 is a transformative force which enhances and incorporates various advanced technologies such as artificial intelligence, machine learning, big data analytics, cloud computing, cloud networking systems, CPS, and much more. So to quickly glance to the previous industrial revolutions, starting from 1700s, the first industrial revolution took place during the 1700s, which initiated steam engines. That is when the steam engine was invented. The second industrial revolution happened during late 1800s, which included mechanization. The third industrial revolution happened during 1800s, during 1900s, which brought into picture the concept of computers, electronics, and also in the invention of internet. Now, today, as we speak and I stand here, today we are in the world, we are in the generation of Industrial Revolution 4, which includes CPS. CPS stands for Cyber Physical Systems. We have Internet of Things, Industry Internet of Things. We have other AI, machine learning, big data analytics, etc. The ones which I mentioned before. So what is the need for this? Transformation into newer things is no more a nice to have thing. Rather, it is something essential, because if we don't inculcate the newer concepts of the eras, we'll be left obsolete in this world. So in order to understand the need for this, I have conducted a survey with the talent acquisition personnel of the industry, wherein I was able to, the rationale behind this study of mine was to understand the behavior and the perspective of the respondents towards Industry 4.0 and the manifestation of Industry 4.0 in our industry, which is the pharmaceutical industry. So this term, Pharma 4.0, has been coined by the organization ISPE, which stands for International Society for Pharmaceutical Engineering. So through this study, I was able to find out that almost significant 80% of the respondents felt that there is a significant positive impact that the Pharma 4.0 will be having in the coming decade, in the coming 5 to 10 years of the future. And then another thing which could be concluded from the study which I've conducted was that about 30% of the respondents have felt that inculcating Pharma 4.0 in our industry will definitely improve the efficiency, will help us in cost savings, and also in improving the efficiency of the production and manufacturing processes. So definitely this deems that encouraging the concept of Pharma 4 and its innovation is a must in today's era. And moving forward, there are various stages. There are precisely several stages to digital maturity in today's Pharma 4.0 in our industry, which is connectivity. We have connectivity, we have predictability, we have visibility. All these stages help us in improving the industry as a whole in reaching to greater heights in the future. Hence, I would like to conclude by saying that the manifestation of Industry 4.0, which is known as Pharma 4. It is a boon to our industry, and also saying that it is a boon to our industry and that this requires a paradigm shift, in terms of manual to a state of automation. This creates a window of opportunity for allowing us to create a workforce for the future, an efficient workforce. In fact, I would say an efficient workforce for the future to come. And then this, as I mentioned earlier, that this will definitely and greatly help the industry in enhancing the production and manufacturing processes and in all outcomes whichever possible. Thus, industry is overall benefited from it. By these lines, I would like to thank the organizers and my institute for helping me and lending the relentless support and efforts. Thank you, one and all.
Yeah. Sorry, was I browsing through the contents of my presentation? No, I was just checking my pulse on a healthcare app I just downloaded. So how cool is that to check? Yeah, yeah, Akash. Yeah. So how cool is that to track your blood pressure, sugar levels, calorie intake by just a click away? So a very good evening to all my fellow presenters and all the respected guests present here. Myself, Akash Anne, pursuing fourth year B.Pharm at SVKMS NMMS Hyderabad. I'm here to present on the topic Artificial Intelligence, a revolution in pharmaceutical industry, recent trends and future prospects. So, apart from using AI in calorie tracking, smart health wearables, etc. So, scaling up this technology of AI into computational algorithms and integrating this into the growing pharmaceutical industry would change the dynamics of the whole industry. So let me continue with talking about the novel approaches of AI being used in drug discovery and development. So the basic or the baseline process in pharmaceutical industry is drug discovery and development. So as many researchers have confirmed that drug discovery and development is a tedious process. It requires about 12 to 15 years and huge manpower and resources, and also a huge investment of $2.1 billion. So what if I told you by using AI tools, the time frame and the resources required could be reduced? So coming to the novel approaches used of AI in drug discovery, AI is used in polypharmacology. AI is being used in drug repositioning to find new uses of the target molecule. And also AI is being used in drug discovery to predict a 3D structure of a protein molecule. So the recent game changer, which has been introduced by a company, DeepMind, is AlphaFold. It is basically an AI algorithm which detects about 90% of the proteins present globally. There are about 200 million proteins present globally. And this AI tool is an open research database which could be accessed anywhere from anyone across the globe. By accessing this, scientists and clinical research specialists could continue with the research work by accessing about 90% of the protein molecules. So continuing with a specific part from the drug discovery, AI use in clinical trials. The basic methodology being used of AI tools in clinical trials is biomarker verification and cohort studies, which gives both a retrospective and prospective perspective, and also clinical trial matching. Clinical trial matching is basically where the AI tools are used to see if one patient is performing one clinical trial, is a volunteer for one clinical trial, is matched with the AI algorithms basically match them and see that the patient is volunteering for only one clinical trials. So these AI, there are many AI companies coming up like Oken, AI, and Cure, which are basically optimizing this levels of clinical trials and help in data storage, data analysis, and optimization of the data. So coming to the recent trends of AI in pharmaceutical industry, there is 3D printing come up. So what if in the future, your local pharmacy store has a 3D printer, and this 3D printer could customize a personalized medication for, just concluding in 1 minute, personalized medication for you. And also there are many uses like drug repurposing or drug repositioning, and blockchain technology where the data is integrated into blocks, and this data is such security that no data is to be leaked, and also it increases the patient adherence level. So summarizing this, I would like to conclude by saying the use of artificial intelligence in pharmaceutical industry, the use of artificial intelligence in pharmaceutical industry could change the dynamics of the industry and put a dent in the industry. And I personally feel that the technology of AI is still untapped, and over the coming decades, it will be unleashed and change the perspectives of the industry. And finally, I would like to thank Narsimha Institute of Management Studies for giving me this opportunity to be able to present here and give me these resources. Thank you.
A very happy evening to everybody. Myself, Varsha from from CMA College of Pharmacy, studying Pharmacy second year. I'm here to present my poster presentation about role of 3D printing. 3D printing is also known as additive manufacturing. It contains several layers under computer program, which is each layer contains a specific instruction. There are two types of methods: direct method and indirect method. Direct method is to design the product by 3D printing, and indirect method is to create a model. Materials are in this 3D printing, materials are there are very less usage of 3D materials, and also it is very cheap, and it contains very less time, and also it contains very less materials. Medical appliances used in 3D printing are bioprinting in tissues and organs, personalized dosage drugs, and unique dosage drugs, and also medical drug profile. Why to use the 3D printing is because in very less expense we can do mass production. By concluding, I can say that 3D printing is used because we can replace or remove the drug, and also we can while coming to children, we can use the drugs on their basis on their interest, like shape, size, color, etc. Thank you.
Okay, right. What's your name?
Good evening, everyone. I am Bhavani from CMR College of Pharmacy, studying PharmD second year. And today my poster presentation is on digitalization of medicine. So digitalization refers to integration of medical knowledge with IT technology to improve the quality of treatment. So the advanced technologies include artificial intelligence, big data analysis, and mission modeling. So due to this digitalization of medicine, we can reduce the manual errors and we can improve the quality of the treatment, and we can reduce the chances of useless appointments. And it is used, and the methods used in digitalization of medicine are apps like Arogya Setu, which is used in the during COVID-19, and electronic medical records, electronic health records, telehealth, telemedicals, etc. And digitalization of medicine is used in other sectors like pharmacy, lab, and in the field of research, and in the field of health sectors. Coming to education, we use like 3D printing, electronic libraries, etc. Whereas coming to hospital, we maintain the records of the patients and also in the billing counters, etc. So coming to research, it is used in DNA sequencing, etc. So digitalization, it is upcoming technology, and it is used to reduce the cost of treatment and to improve the quality of the treatment and to store the records of the patients. Thank you. No, ma'am, we are nervous, ma'am. Yes.
A very happy evening, everybody. Myself, Kovida. I'm pursuing PharmD second year of CMR College of Pharmacy. So my topic is about ingestible body sensors pills, ma'am. And so, as we know that in nowadays in healthcare sector, so ingestible and swallowable pills it is used for the patient monitoring and patient counseling, and also these are for used as a drug delivery applicants. So coming to the NDA species, like in the year of 1957, the first pill was made up by radio frequency. And the year of 2017, it was made up by digital pill. And in the year of 2018, heme-sensitive probiotic biosensor came into existence. And recently, they have brought this up of ingestible body sensor pill, which is used for the GIT track identification if any disorders are present in the GIT. So coming to the work of how this biosensor pill is used in our body. So we'll ingest the pill and then it transfers from the esophagus and to the stomach, and then it goes into the rectum. So how in the stomach, there are many enzymes present in our stomach. So this also contains a sensor, and this also contains a biomarkers in it. So because of that biomarkers activation, we can know the disorders if any present in our stomach. So these are edible microchip sensors, ma'am, and also these are activated by stomach enzymes, and we'll keep a patch on our skin. So on activation of biomarkers, these enzymes will release receptors, and that will send the signal to a phone app. So this is a mechanism of ingestible body sensor. So coming to the materials and methods which are used in the sensorable pills are IMBED, which is used in the identification of GI bleeding. So this IMBED full form is indigestible micro bioelectronic device. So this is also having heme sensitizing, which is having a bacteria like lactobacillus and also Escherichia coli. These both will used in the outer covering of the pill. And also we have a no, let me give a conclusion. So conclusion is this is used for the maintenance of pH, temperature, and also it is used for the pressure control and also for detecting the GIT problems in our body. So these ingestible body sensors are used very warily in our country nowadays, not in India, but out. But it is coming into the review in India nowadays. So this is very useful for the geriatrics, not pediatrics more. More geriatrics are used for this indigestible pill. So I'll conclude by this. Thank you for giving this opportunity.
మేడం నేమ్ చెప్పారా?
పాస్వర్డ్ ఏంటి మ్యామ్ తో ఫోటో ఇదిగో అక్కడ మ్యామ్ తో దిగుతుంది ఫోటో తీయండి తీయండి ఇదిగో నీవే all the best.